Good morning, everybody. Thank you so much for being here. So this is what's the actual title of our session? Anybody have it? We don't even know the title of the session. I'm glad you're here, but we don't know what we're going to talk about. Jeff, you have the
Cancer, Pharmacoepidemiology, and Genomics. What you should know in the era of precision medicine.
That's the title, and this is the 13th year we're actually giving this course. The last two were online, and so at least my lecture will be a little bumpy. This is the first time I've given a lecture in person in 4 years. So it's not on Zoom, so if I do some weird stuff, just no. Let me start out by introducing our faculty. I'm Andy Friedman. I'm with the National Cancer Institute. And Donna over there, she'll introduce herself later, but she's from the FDA. Donna Rivera, and Wei Zhou is with Eli Lilly. And Jeff Liu is with Prince Margaret Hospital and a lot of other hospitals and universities. And is Bruce there? Bruce just walked in. Wave Bruce Carlton from Children's British Columbia. So we put together a really nice group of lectures, and we will have a interactive panel discussion afterwards. And so I think most everybody's here, so we'll get started. So the talk that we're starting with is designing and analyzing pharmacoepi studies in cancer research. And so this is actually feedback we got from many of the students who took this course before, that they wanted a lecture on cancer risk, medications and cancer risk in cancer. And so a lot of what I'll cover is basic pharmacoepi, but with a little bit of a cancer flavor. So just move on to the and so these are the talks that you'll hear today. So as we all know, pharmacoepi and pharmacogenomics can play important roles in a variety of cancer-focused studies. And so in this afternoon, we'll be discussing pharmacoepi in the context of cancer drug association studies. That's the talk I'll be giving. And Donna Rivera will be talking about real-world evidence and data sources. And Wei Zhou will be talking about oncology drug development. And Bruce Carlton will be talking about studies of adverse event related to cancer therapies. And then Jeff Liu will talk about population-based studies of oncology drug effectiveness. But before diving into those, I wanted to give a very quick review of some of the important cancer biology to keep in mind while thinking about the design of these studies. One of the most important when talking about genomics is that when we're talking about cancer, we're really talking about evaluating two genomes. First, you have the germline genome. This is everything you've inherited from your mother and your father. And if you work in the pharmacogenomic space outside of cancer, it is probably what you're familiar with. It's very important in predicting drug exposure and toxicity. In cancer, it can help optimize the efficacy of treatment. And so the types of things we're talking about is like BRCA mutations. Usually we talk about that in relation to cancer risk. But basically we're referring to inherited germline mutations. The second is the somatic genome or the tumor genome. This is all of the acquired genetic mutations that have developed within the tumor. It can be very important in predicting response to therapy and as disease markers of cancer. For example, BRAF mutated melanoma is defined by a mutation in the gene BRAF that is present in the somatic genome, but is not necessarily present in the germline. So all of these mutations come into play when we are discussing carcinogenesis. And as we all know, cancer biology is a complex system of molecular signaling pathways and biological changes. The multi-stage process of carcinogenesis, which includes initiation, promotion, progression, invasion, and metastasis, corresponds with specific mutational or epigenetic changes causing biological changes within the cells. Endogenous and exogenous substances can initiate DNA damage, and these initiated cells can then begin clonal expansion with promoters that can help speed up the process. So there's a lot of etiological factors that can drive carcinogenesis. They can be genotoxic, which implies causes direct DNA damage. And these are things like radiation, UV light, or reactive oxygen species. They can also be non-genotoxic, that do not directly damage the DNA, but disrupt biological processes like the immune system, inflammatory responses, or endocrine systems. And in this case, we're maybe talking about medications that we want to look at increasing or decreasing cancer risk. So these changes within the cell affect tumor suppressor genes or oncogenes that are important for regulatory processes like cell survival, genomic maintenance, and cell fate, all of which can confer a selective growth advantage and lead to the development of cancer by bypassing the normal checks and balances present in our cells. We know that there are medications that can affect these cellular processes at any stage, increasing or reducing the risk of cancer. Well-designed pharmacoepi studies can examine the relationship between medication use and the risk of either initiating cancer or promoting cancer. So having an understanding of the biological mechanisms and these disease pathways can provide complementary knowledge when forming hypotheses, designing studies, and analyzing cancer as an outcome. Although our gold standard, of course, is clinical trials, we all know that observational studies can capture the real world use of patterns in larger, more representative populations for longer time periods. But when we're talking about cancer risk and medications, you can't ethically do a trial where you randomize people to a medication and not a medication and look if it increases the cancer risk. So in most cases, we're talking about observational studies. Now, there have been trials of protective agents that try to reduce your cancer risk, but not in the case of if we're worried about a increase in cancer risk. So with this in mind, we're going to walk through the considerations for designing a pharmacoepi study of drug cancer associations and discuss the cancer biology when relevant. We're going to use a case study to put these considerations in context. And the case study is, do PDE5 inhibitors increase the risk of melanoma? And PDE5 inhibitors, what they are used for erectile dysfunction. So we're talking about Viagra and Cialis. And do those actually increase the risk of melanoma? So we're going to do a very high level walk through these considerations. I will put some resources at the end if you want more detailed information, but basically we're going to start with data sources, go through biological plausibility, study design, exposure ascertainment, and outcome ascertainment. And this is no different from any of the usual pharmacoepi studies that you would design. There are just some little differences when it comes to cancer. So let's start with data sources. Most of you know all of these things that I'm going to say in this slide, but we'll start with data sources. And of course, there are several types of available sources, including administrative claims data, EHRs, survey data, and disease registry data. All of these have advantages and limitations. Picking the correct source will depend on the type of data you need. For example, some may include dose of medications, while others only include that it was prescribed. Not all sources will have the same data elements collected, and there'll be more detailed information on data sources that Donna Rivera will give in her talk. So for our case study, we have access to two data sources, the Danish Nationwide Health Registries and Kaiser Permanente Northern California Health Maintenance Organization. The Danish registry is population-based and covers all Danish inhabitants. We can use a linkage that includes pharmacy data and cancer information. For our study, we have the type of drug, the data filling, and the quantity. We will have incident cases of cancer based on ICD-10 codes as well. For Kaiser Northern California, which is an integrated healthcare system, it's in the US, this data is specifically from Northern California. We have medication data that includes the date of dispensing, the dosage, the day supply, and refill information. The cancer registry data includes diagnosis date, cancer site, histology, stage, and treatment. And these are both really good sources of data. However, we still would not have data on adherence or frequency of use of PDE5 inhibitors. So since we're studying melanoma, we will have data on other diagnoses that may be important for the risk of melanoma, but we will not have access to any information or dates concerning sun exposure or sunscreen use. So those are limitations, of course, to keep in mind. So we have our research question and our data resources and our data sources, but there is is there potential mechanism for carcinogenesis? PDE5s, as I mentioned, are used for erectile dysfunction. So how would that impact skin cancer risk? For this, we need to think about the biological mechanisms involved in the cancer biology and mechanisms of action of the drugs. What we do know is that half of melanomas have a mutation in BRAF, which suppresses PDE5A. This suppression stimulates melanoma cell division and metastasis. So PDE5 inhibitors work in a similar fashion in that they also inhibit PDE5A. So there is biological plausibility that pharmacological inhibition of PDE5A may also increase the risk of melanoma. So our next step is to decide on a study design. Here we have the traditional pyramid of evidence quality. But for our pharmacoepi studies, we're going to focus on cohort and case control studies for this case study. As I mentioned before, RCTs are usually our gold standard, and observational studies can provide a lot of complementary information. But in the case of cancer risk, we're really mostly talking about observational studies because it's not ethical, once again, to do a clinical trial. So this is basic pharmacoepi. So cohort studies are comparing users to non-users or an active comparator and prospectively following them over time for the outcome of interest. In this case, a cancer diagnosis. This can be efficient when exposure is rare, but the outcome is not rare among users. Additionally, if you're planning on looking at other outcomes in the population, having an available cohort study is helpful. Cohort studies are also useful if you need to look at new users versus prevalent users. You're also able to use an active comparator in this design, which may be important if users of the class of drug would be very different from non-users. Cohort studies often need much larger population sizes compared to other study designs, especially if you have an outcome of interest that may be rare, in our case, melanoma. So in case control studies, you're comparing people with the outcome of interest and those without with respect to their history of drug use. These are generally retrospective and can be nested within a cohort study. Because cancer is a rare outcome, it can be useful to have a population that already has the outcome of interest. This is also a useful design if other outcomes from medication exposure are going to be assessed. Taking into consideration our data source and retrospective nature of our question, we'll be using a case control design. In both data sources, the Danish and the Kaiser, we will match men that have a history of melanoma and assess their PDE5 inhibitor use. To be able to define our cases and controls, we will need to decide how to define our exposures and our outcomes. So first, how will we be capturing dose information? Thinking about how the drug is used and the pharmacology can be useful, whether it is an acute high dose or a chronic low dose. What is the mechanism of action? Is it affecting an important pathway? Is it causing inflammation? What is the half-life of the drug? Does it build up in certain cells or is it cleared quickly? All these biological and pharmacological characteristics can help in determining the appropriate way to quantify drug exposure in the study. Most known cancer drug associations are dependent on cumulative amount, that is, stronger associations with higher doses and longer-term use. You can quantify drug exposure by using cumulative duration of use, cumulative quantity of the drug, or prescribed daily dose coupled with duration of use. Your options, of course, are going to be heavily dependent on the data you have available. For us, we have prescription information that includes the number of tablets, so we can use that to estimate categories of cumulative use. We also need to define our exposure window. We know that there is a period of time in which a person is susceptible to the effect of the exposure. From the first occurrence of a cluster of cancer cells to a stage detectable by screening measures or clinical symptoms of cancer is typically very long and could be 20 to 30 years in some cases. It is very unlikely to have a cancer diagnosis shortly after drug initiation that is related to the exposure. There are exceptions, of course. And finally, drug use has been shown to increase in the year prior to a cancer diagnosis, most likely to address the symptoms of the underlying cancer. A really good example is of those of middle-aged man who comes in for obstructive urinary symptoms and is prescribed a drug to relieve the symptoms. After additional tests, he's diagnosed with prostate cancer. So just looking at the data, it looks like there's an association between the drug and prostate cancer. However, it was treating the symptoms of an undiagnosed prostate cancer. So our goal is to collect a study period that captures the relevant time period of exposure and allows for adequate time to detect cancer as an outcome. So that brings us to lag time. So in our study design, to counteract the latency time and any possible reverse causation, which I just talked about, we will introduce a lag time. So that's the optimal period for addressing the long induction and latency periods. And that will depend on the specific drug cancer association. For example, there are some rapidly developing cancers like non-Hodgkin's lymphoma after immunosuppressants are given to transplant patients. So that can happen very fast. But for most cancers, the development is usually much longer. And so concerning the effect a drug has on carcinogenesis, if it's an initiator affecting the early stages, the risk window may not start until long after the initial exposure. Whereas if the drug exposure acts as a promoter, the risk period could be much closer to the drug exposure. Of course, there's going to be uncertainty around what the correct and biologically relevant lag time is, but you can evaluate various lag periods using sensitivity analysis. So for our study, our primary exposure will be 2 or more filled prescriptions of any PDE5 inhibitor. We will have access to prescription data, including the number of tablets, so we can categorize cumulative use. We will introduce a lag period of 1 year prior to diagnosis. This should cover latency as well as any reverse causation, although unlikely with this drug cancer association. Finally, we need to decide on how we're going to define our outcome. So the validity of your outcomes are going to be very much dependent on the data sources due to where they receive their diagnosis. Completeness and validity increase with the number of modalities, such as medical records and path reports used to ascertain the outcome. So we need to determine what specific diagnosis we want. When thinking about our biological understanding of cancer and its subtypes, it's very unlikely that a drug exposure would increase the risk of all cancers. And keeping all cancer as an outcome would be difficult to interpret, and is usually driven by one specific cancer with the highest incidence. So we've discovered a lot of mechanistic underpinnings of cancer development, and we've shown that different disease markers and molecular features that not all cancers within even the same site have, and they all have different characteristics. So if this study were being done today, this study was done in 2012, we would have a lot more information on the biology, the molecular markers within the tumor. In this case, we're really looking at the histology. So our cases will be defined as men with histologically confirmed melanoma between the ages of 18 and 84 years of age. The date of diagnosis is the index case, and we're going to match 10 male controls to each case by birth year, applying the same exclusion criteria as for the cases. In the Kaiser population, we will also match on the length of continuous health plan membership to ensure similar availability of medication use and other health data. Please note that in the real study, there's a whole host of other exclusions based on other diagnoses and conditions, but for simplicity's sake, we're focusing on cancer diagnosis. So just reviewing for our case study, does PDE5 inhibitors increase the risk of melanoma? The data sources were using a Danish data source and the Kaiser. There does seem to be biological plausibility of pharmacological inhibition of PDE5A. We're going to use a case control study design. We're going to use for exposure ascertainment, we're going to use a 1-year lag time and use categories of cumulative use based on the number of tablets. And for the outcome ascertainment, we're going to use histologically confirmed melanoma diagnosis. And so what we've basically done in this lecture is described a study done by Dr. Anton Potegard, which was published in the British Journal of Cancer in 2016. He originally gave this lecture many years ago. In that study, they identified over 7,000 cases of invasive melanoma for the Danish registry and another almost 3,000 cases from the Kaiser Northern California data. And in the end, they did not find any evidence for a causal relationship between PDE5 inhibitor use and the risk of melanoma, which is really important since these drugs are really highly prescribed. I want to again point out that this was a very simplified, high-level version of designing a study, just to start the discussion of the factors one would consider when designing drug cancer association studies. We did not discuss potential issues of confounding, and as in the case of any study, we're unable to control for everything. The paper shown here, again by Dr. Potegard, is a great resource and also includes a table at the very end that lists, I think, 10 different studies of interest that include methods as well as cancer biology related to this topic. I hope you'll take a closer look at those in more depth if you're designing any of these studies. Let me just mention that this is another review paper that actually came out of a workshop that the FDA gave in 2016 and highlights a lot of the concepts that were present here. And there's one other paper that I wrote with my colleague, Dr. Rivera, on the relationship, understanding biological mechanisms and how it relates to pharmacoepi. It's another really good resource. And I think with that, I'm going to stop. And I think we might have time for just 1 or 2 questions. Otherwise, we can take questions at the end. And yes. Yeah, I mean, my answer to that is, it's you have to kind of do sensitivity analysis. So they had no use, they had 100, there were 300, there were 500, and they broke it into different categories, and they were looking really for a trend. And really interesting is that they did find an increase related to these drugs of in situ melanoma, very early stage melanoma. But they really attributed that to the fact that these men are going to the doctor, they're getting their prescriptions, and they're being more closely monitored, and they might see a very early melanoma. And so those are all types of things you have to take into consideration. But what is the right dose and cumulative dose to look for that? I think it's really a sensitivity analysis, and it depends on the data that you have. And yes. Yeah, they might not be able to hear you.
People in the back probably can't hear the voice. Sorry about
Just a
that.
question. Thank you for this nice example. Well, I suspect for some cases it would be. Can you hear me?
Is the mic working?
Maybe like no.
Just hit it. Just hit it. Right now. Right now.
La la la.
No, it is working.
It is working. So the question is, in some cases, I suspect it's important to collect the information which is not in our databases when we have a strong risk factors related, let's say, to lifestyle. Do we have recent examples of some more modern designs with the data were collected for both study participants and in the databases?
So is your question for, in the case of melanoma, we don't have data on sunscreen use or lifetime use of sun exposure, right? And those are major risk factors. And your question is, what do we do when we don't have that?
Yeah, or do we have some recent examples when people tried to do something with this?
Yeah, I don't know the answer to that question. Is anybody else? I'm not sure of examples. In some cases, you can, you know, especially with the cohorts, if you are doing propensity score analysis, you're hoping that your cases and controls are similar on those risk factors. It's probably unlikely that men who have erectile dysfunction got more sun. It's possible, but it's possible, but you know, you have to make some assumptions, and you know, you're limited by the data that you have. You know, but there are other large epi studies that do collect lifestyle factors like sun exposure, but then they don't have the drug data. And so it's a difficult thing to measure. So I'm not sure I have a good answer to the question.
Thank you very much.
Thanks. So I think we can move on. And all yours, Dr. Indigo.
All right. So good afternoon, everyone. It's really nice to see you. It's really great to be here in Halifax. So welcome. I am Donna Rivera. I'm the Associate Director for Pharmacoepidemiology in the Oncology Center of Excellence at the US FDA. And today I am joined by my colleagues here to talk a little bit more about cancer pharmacoepidemiology. And I'm going to focus on oncology drug development from a regulatory perspective. I have nothing to disclose, and the views presented here reflect my own and not those of the US government. During the presentation, we're going to try something out. This is a new software the FDA has for polling. And if you're able to participate, I would really appreciate it because we are testing this for the Friday plenary. So it would be good to make sure this works this time. So while you get this squared away, I'm going to start this session. It looks like the / the session ID, the last 6 digits are actually going to be slightly different. Does this work when you do this, when you log in? Does anything come up? It does not. Okay.
It does not exist.
So it's 287453.
Sorry, can you say that again?
Sure. 287453. Okay. Great. All right. So we're gonna see if this works. Hopefully. Okay. So I wanted to just start out by seeing who's in the room. And hopefully this question appears. Is this on the software or do you see it? Oh, sure. 100%.
Except the last 6 digits or something.
Right.
287453, right?
Yes.
And yes, once you log in.
It's good.
It's good, except how do you click on the answer?
It shows the question.
And does it show?
I mean, it shows the question, but what do you enter?
It says
Well, that is-
participant messaging has been disabled for the session.
Oh,
Is that what it says?
that's
Yeah.
good.
If you click on the little text thing.
Well, that's really great.
Your instructor has ended the session. Is
I'm
that you?
glad, yeah, because I'm gonna try to figure out, it appears that our IT department maybe didn't set this up exactly right, so I just wanna see if there's, I appreciate you guys testing this out for Friday, because this is something that we definitely want to work for that. Let me see. Technology is always fun.
And the session on Friday is nothing important. It's just like what the plenary, right?
Yeah, that's right, so.
Not really important. It's just a plenary.
Yeah. With the Health Canada and FDA leadership, yeah, I feel like we definitely want that to work. Let's see. What I might just do for the purpose of this session is just go through, and if you can just raise your hand, we'll just do that for now, and we'll try to get this IT situation worked out on site. So
So you're gonna show these.
yeah, so I mean, it has this feature where you can theoretically poll, but obviously that is incorrect and does not appear to be anything that anybody answered. So for the purpose of, I just want to get a sense of who's in the room. So what is your primary career sector in pharmacoepi? So if you could just raise your hand, academic. All right, fair amount. Industry. Government. All right. And then CRO. Okay, other. Pretty much everything. All right, so it's a good meld of different groups here, a lot of academic and industry. So hopefully this talk will speak to those needs in a variety of perspectives. I'm going to start by discussing the landscape of real world data types. I'm going to discuss the strengths and limitations of some of the different databases and data sets for RWE studies. Then I'm going to illustrate some methods for oncology real world data design and analysis. And then finally, I will highlight collaborations in real world data. So I've organized the talk into 4 sections: real world data, epi methods, regulatory review, which I think is kind of an interesting lens of pharmacoepi in the context of real world data, and then novel designs. So we'll start with real world data. Here's another poll. This is a little challenging to actually ask. in this particular context. So we're just going to say, for the purposes of this, I assume that most of you are interested in oncology, but we'll touch on all different, pretty much all of these in some way or another throughout this talk. All right. So we're going to start very regulatory in nature and use the FDA RWE framework to provide definitions for real-world data and real-world evidence. Real-world data are data relating to patient health status and or the delivery of healthcare routinely collected from a variety of sources. And real-world evidence is the evidence about the usage and potential benefits or risks of a medical product derived from analysis of that real-world data. And so thinking a little bit about the real world data sources and types of data, I think about it in this construct, which is that the sources include items such as electronic health records, registry data, which is there's a variety of examples of this in the US and internationally, programs like SEER that are organized systems uniformly collecting clinical and other standardized data to evaluate outcomes, patient-generated health data, so that's patient or family-generated data, and then administrative claims, which is typically submitted by insurers for payment. And within the context of the sources, there are a variety of real-world data types. So within each of the sources, you can have information such as demographic and social information, pharmacy information, radiology information, and patient-reported outcome data. The FDA does not endorse any one data source over another or seek to limit the possible types of data sources that can be relevant to answering study questions. Thinking a little bit more about the strengths and limitations of these data, I think it's helpful to think about, of course, what your study question is first, and then thinking about the strengths and limitations of the data set. So in electronic health records, you have longitudinal, very granular, detailed picture, including potentially unstructured data. The limitations, though, are in completeness of capture, because it's going to be limited to capture within the system. Enrollment and care relies on physician entry and is very clinically demanding for most physicians to do. And so that data that's input is the data that is output to you as an analyst. So therefore, it can be analytically challenging as well. From a claims data perspective, again, you have that longitudinal but you have defined enrollment, which is a difference, I think, in the context of knowing typically when the start and stop of an enrollment period is. All billed care is captured. But again, you have the same issue in the completeness component that it's only captured by source. So it lacks the detail of, for example, something like PD-L1 test results may not be available in the claims data. From a registry perspective, you have longitudinality again. It can be population-based depending on the specific registry of interest, but often the larger registry programs have data delays, and there are, because of the type of, or the way the registry is set up, it may depend, but there can be limited availability of specific clinical data elements or treatment data that may be relevant to your study questions. Important to think about that. And then patient-generated data is data generated directly from the patient experience. So it's highly detailed, and you really often can't find it anywhere else in any other data. There's some interesting and novel digital collection strategies, so really ways to create patient-centric generation of data. However, the quality is variable and depends on how it was set up, and measurement in this context is very important. Measurement is always very important, but I think in that particular context can be quite important. So thinking a little bit about structured and unstructured clinical data, in the context of mostly structured data, as you're thinking about your study, how you're going to design it, and what the data elements are, the lab values, pharmacy claims, demographic data, they tend to be regimented into specific selections, right? I think of these like drop-down menus, right? You have a specific input. And then on the unstructured side, often things like pathology, genomic data, they're in PDF, they're in all different types of formats that you may or may not be able to directly extract. So that makes your data cleaning and your data input much more challenging on the upfront. And then in the middle, medical oncology records, patient-reported outcomes, they can be structured, they can be unstructured, it kind of depends, but it's really important that you know your data set really well and you know that information upfront so that you can figure out exactly what you'll need. and so that you can make sure that you have the data that you need to conduct the study. For example, if you're going to do a study looking at EGFR mutations in non-small cell lung cancer and you don't have test result data, that could be quite a problematic and challenging study question to answer with your data set. So in the context of an example, give an example of looking at a bunch of different types of data and how you might use them. This is an example in breast cancer. So you may want to obtain this type of information about receptor positivity or histology from the pathology data. You might want to obtain the pharmacy medical product data from the pharmacy data. From imaging, you may be interested in different types of imaging that occur depending on what your endpoint is going to be. You may be interested in looking at response. From radiation oncology perspective, you may be interested in getting additional information. Genomic, there's a variety of different types of genomic markers, genomic tests that you may want to look at. Comorbidity data that may or may not be important to your study question, depends again on what it is. Your procedural data and patient-generated data, things like ECOG status can greatly impact the study. So looking at all these together, the goal is kind of how do you think about what your study question is and how it ties to your data set and the elements you may need in the totality of what's available. So thinking about the totality of what's available and how this might be challenging, there are challenges to use of oncology real-world data, and those include the complexity of cancer care, expanding data characterization around each cancer, so the evolution of precision medicine, dispersion of diagnosis and treatment across a fragmentation of healthcare providers, and the changing demographic distribution of the aging U.S. population. So now we're going to move to a brief bit on epi methods and talk a little bit about this in the context of real-world data. So I think I've said it, and I'll continue to say it and say it again, but this is really the beginning. Is what is the research question? So what are you asking, and then how can you answer that question? I think this is so important because this is just a graphic really showing that the majority of the data that we use in the context of observational studies is secondary data. So it's reused, it was collected for another purpose, clinical, administrative, and less likely, there are obviously examples, but less likely to have been collected specifically for research. And this comes into play when you think about things like how pharmacy data goes through an electronic exchange. So the prescriber orders a medication or there's a MAR, then the pharmacy receives the claim. They may adjudicate the claim. There may be a rebate. It might be cash pay. And the pharmacy adjudicates the claim through typically a switch vendor that receives all claims. So if you're using switch vendor data, you would have non-adjudicated and adjudicated claims, which does not necessarily mean that the patient received the prescription. And then you have insurers that have all paid claims. So the difference here is that if you have the insurer and you're looking at this kind of whole, prescriber, pharmacy, switch or intermediary composition, the insurer will have all the paid claims, the pharmacy will have the cash pay and rebate and adjudicated claims, and the switch vendor will have all claims, so anything that was submitted. So you can also think about things like looking at access issues by using that data for claims that were submitted, but then were not approved or picked up. And then from a data selection perspective, there are a variety of types of data, but focusing in on claims and EHR data, claims data, again, comprehensive data across all settings, specific to that, typically insurer, and has a lot of information. However, it may miss some clinical details like lab results or genomic testing, whereas EHR data is going to have detailed data within that specific system. But if a patient crosses EHR systems, there will be challenges in the context of being able to effectively use that data. And then thinking about how you create your cohort and you think about, what is the study question? How are you moving towards that? What are you asking? This is an example from COVID, and it's thinking about what the patient population would be for a study. Are you interested in looking at a subpopulation? So for example, patients with cancer. And then in that context, do you also want a non-cancer comparator? Then from the next level, thinking of, do you need testing? Do you need patients with a positive or negative COVID test? are they going to be patients that were on active cancer treatment or not on active cancer treatment? And then what is the modality of treatment that those patients were utilizing? So as you work through your attrition diagram to figure out what you're going to ascertain in your question, then you start thinking about, okay, if this is my question and this is the population I want to ask it in, how do I get to a data set that's going to be able to effectively answer the question? So this really comes down to the context of what the target population is. And from a regulatory perspective, this is often what is the intended use of the medical product. Then you have the study population. That's the cohort you've selected from the available data in the analysis population. And then there is, of course, what you are drawing from in totality, which is the broader population. And this speaks to the concept of generalizability. How does what you've selected in your study generalize to the broader population? So here's another polling question that that our polling software is a little challenged by, but I just wanted to pose this, and I think that E is obviously, you know, not a real answer, but the question which I would have been interested in is, and maybe we can just do it with hands just to see what people say. What do you think is the most common or important methodological issue with real world data? Selection bias. No? Confounding. Okay. Missing data. All right. Immortal time bias. All right. There are no specific issues. I guess you could look at this both ways. There are no specific issues because there are many specific issues, but yeah. Yeah, that's it. That's it. Well, I think that I was interested really in what people think is the most pressing issue. Well, okay, so I mean, to be honest, I think it really depends on the study. It depends on how you set it up, what the most pressing issue is. The real answer, though, to your point is really a little bit of everything. It's kind of all of them. So potential challenges with retrospective real world data in generating evidence of treatment benefit. I think when you think about the bias, the confounding, the potential methodological issues, you have to think about why you have those issues. And the challenges are the lack of randomization when we're talking about many types of observational studies, but hold that thought because I will talk a little bit about randomized observational studies in a little bit. The lack of structured data and need for extensive curation, the inability in oncology to collect tumor-based endpoints like response, completeness of data capture, and data quality and missing data. I'm going to focus on three because otherwise this could be a very long talk, and which are really thinking through the different factors that are methodological concerns with the use of real data, and those are selection bias, confounding, and missing data. So we're going to start with selection bias. And selection bias is a systematic error in a study that occurs due to factors that influence study participation or eligibility. So it's this distortion of an effect that arises when the association between the exposure and the disease differs for those who participate and for those who do not participate. So this could be due to a variety of factors that can induce potential selection biases. And so when thinking about your study and thinking about the entire population that you have, and then the data that you're using, it's important to think about, do those reflect any type of selection bias? The next is confounding, and I think this is something that is just a classic pharmacoepidemiology, and I'm sure that all of you know, but you know, as a confounder, we think about a confounder. A confounder is a risk factor for the disease or outcome associated with the exposure, and then is not on the causal pathway. So it must not be an effect of the exposure. And it's a distortion of the measure of the effect of a medical product on an outcome due to another factor, not on the causal pathway that is associated with both the product and the outcome. So why are we concerned about this? We're concerned because a distortion of the relationship can cause a false or spurious association when not appropriately controlled for in a study. And so this is it. Lack of randomization leads to a much more significant concern and potential for confounding. And in Section 314 in the Code of Federal Regulations, where we look at a lot of what we do in the context of adequate and well-controlled studies for FDA review, this is actually mentioned in a certain way that in establishing effectiveness, it's essential to distinguish the effect of the drug from influences such as spontaneous change in the course of disease, placebo effect, or biased observation. So we're looking carefully for this. And there's obviously a myriad of ways to address confounding, both measured and unmeasured confounding, and a great paper by Schneeweiss that talks a lot about this. And of course, careful adherence to GPP can help to minimize bias and confounding. So from a study conceptualization standpoint, it's also, as I've been discussing through this, thinking about your study question, your study objective, and how all of these aspects fit together. So whether your objective is effectiveness, a labeling change, post-marketing, or contextual evidence, thinking about the key design elements of your data source, your study population, exposure ascertainment, outcome, and covariate ascertainment, which were mentioned by Dr. Friedman as well in the previous presentation. So it carries through that these design elements are really important. And I would emphasize that in the context of regulatory pharmacoepi, that you speak with the FDA for those of you that are in industry early and often if you are looking to use real-world evidence in your study. And that's because of the potential uncertainty in the context of evaluation in the absence of randomization, right? So this is where the concerns around bias and unmeasured confounding and certain other contexts lead to the potential for uncertainty versus the randomized controlled trial, which is seen as the gold standard. Missing data is also a concern, absence of data elements that were either not observed, collected, accessible, or analyzable within the data source. Missing data comes in a variety of ways. So, it can be missing at random, which I think is pretty rare, but missing at random or in various other contexts. But the clinical relevance and importance of the variables and the degree of that missingness is what really matters. And missing data is actually not unique to real world data. There's also missing data in clinical trials. And another component of when we're thinking about generalizability in the current context of our methods, it's an important consideration because it pertains to being inclusive of populations and reflecting the epidemiology of the disease. And from a regulatory standpoint, it's important that it not only is generalizable, but it's generalizable to the US population to meet our regulatory standards in the US. So this is an important context of thinking about when you're setting up a study, what your intention is. If it is not regulatory, then that may not be a factor to you, but thinking about how the study that you do, or even the trials that are ongoing, if you want to incorporate real-world evidence, how that's generalizable to the US population. Some new, I would say newer ideas coming out of the FDA in the context of representativeness and generalizability come from our commissioner, who is really pushing forward the goal of achieving greater diversity as a key focus of the FDA. And I think this is where real world data has the power to advance health equity in creating ways in which data sources are more reflective of the rural population and enhance diversity and inclusion in drug development programs. The other aspect that I think is actually fairly new in the context of review is auditability. So data must be auditable in either source records or certified copies for review if it's being submitted in a regulatory context. And this all falls on the sponsor to be responsible for adequate processes in place. But it's important to think about even when you're doing research, because I think decision-making, whether it's regulatory, clinical, or otherwise, should be built on sound evidence. And thinking about the data provenance, the curation processes, and all that goes into your analytics as you are focused on maintaining data integrity and reliability throughout your study. So now I'll talk briefly about regulations, which is everyone's most exciting topic, but I'll try to keep it interesting and quick. So real-world evidence is actually, in the context of the FDA, is actually fairly new. So we obviously, all of us know that observational data and pharmacoepi has been long-standing for decades, and really in the area of safety has been in the regulatory space for quite a long time. But as we think about its pertinence to effectiveness, I would say that that is the area where it's definitely newer. And a lot of that is related to 21st century cures, which focused on evidence development and modernization, and that in the context of real world evidence. So thinking about the use of RWE for approval of new indications and post-approval study requirements. And then also our Federal Food and Drug and Cosmetic Act. And so that's what is responsible for establishing substantial evidence with adequate and well-controlled investigations. And the goal is to work through a structured risk-benefit framework in the process of a new drug approval so that it can be fairly and responsibly concluded by experts that the drug will have the effect it purports to. And so this is the systematic approach that regulators use consistently to approach decision-making when it comes to medical product approvals. And I will say that it is the same for clinical trials and real-world evidence. So we focus on the use of the regulations for our review process, and that is long-standing. But as the use of RWE has been increasing, the FDA has been responsive to drug development needs through a variety of guidances that have been released, led by the Office of Medical Policy in collaboration with CDER, CBER, and OCE. And the guidances are really useful information, and I've broken into kind of three categories. There's two guidances that focus on RWE and RWD sources, so electronic health records, medical claims, and registries. There are three that focus on the submissions process of real-world data. And then there's one that is our most recently released guidance that focuses on design, and it's specific to externally controlled trials. And I will briefly touch on that as well. In the context of use of oncology RWD for effectiveness, I think it's important to mention that randomization is important. So when you can randomize, you should randomize. And then in instances, for example, when there are feasibility challenges, ethical concerns, or questionable equipoise, it may be appropriate to use real-world data to answer the study question. And so some applications of that are in pediatrics, rare diseases, areas of significant unmet medical need, molecular subgroups, and underrepresented populations to advance health equity. And so from a conceptualization standpoint, as I've mentioned, really the suitability is around defining your study objective and key design elements and determining if the data are fit for purpose, which means they're relevant and reliable to the study question and to the study that you are conducting. And then if so, creating a detailed study protocol and robust statistical analysis plan a priori are important as you move through your study to ensure the integrity of the study. From a perspective for regulatory purposes and use of real-world data, this is a synopsis of many of our guidances and kind of the process that I think is very useful. So predefining all essential elements of the study, describing how the elements were ascertained, addressing any issues to determining reliability and relevance. And that's in the protocol and also includes provisions for ability to audit. And then, of course, consulting with the FDA. So consulting prior to the start of the study if there's any questions. So I will conclude by just talking very briefly about novel designs and different ways to think about real-world data. And I'm going to start by talking about where I think the possibilities and challenges are. And so I think that in the possibilities, we've seen the use of real-world data for emergent public health needs, and it's been, I think, quite an impressive. There have certainly been some road bumps that all of us have seen, but a way to try to respond to public health needs. Novel designs, so external controls and pragmatic trials, the ability to ascertain real-world response, and the understanding of drug effects in underrepresented populations. I think the challenges that are still existing are in the characterization and quality of real-world data, in causal inference, and in the interpretability of time to event endpoints in single-arm studies. So with that, I will talk about externally controlled trials. So externally controlled trials are an approach to evidence generation, and I would like to start with just a definition to keep us all on the same page. So an externally controlled trial is one in which the control group consists of patients who are not part of the same randomized study as the group receiving the investigational agent. In other words, there is no concurrently randomized control group. You can comprise externally controlled trials through a variety of means. There are different sources. Real world data is one of them, which is why we're talking about it here, but you can also use clinical trial data or literature-based approaches. From a temporality aspect, you can have a concurrent or a historical control. And the purpose is typically one of two items. So as a benchmark, as a natural history or classic epidemiology study, or as a comparator, which typically would require individual patient level data, and the focus is on effectiveness. And so in a situation where there are limited or no randomized studies available, it's so to this point so far been limited to providing important clinical context, has been exploratory and considered supportive. A drug has not yet been approved in oncology using an externally controlled design due to various limitations. And so those include the lack of a pre-specified protocol or other issues around lack of formal statistical comparisons, appropriateness of the data source, confounding, which I think everybody noted was an important methodological issue, as well as another very important issue in externally controlled designs that we talk about in the guidance, which is around index date selection and immortal time bias. And then the last project that I will mention is a new project that we've just launched in the Oncology Center of Excellence, which is Project Pragmatica. Project Pragmatica seeks to advance evidence generation for approved oncology medical products by exploring innovative trial design approaches that introduce functional efficiencies and patient centricity through integration with real-world clinical practice. And I'm going to go actually to this slide to just talk a little bit about the intention of pragmatic trials, which is the intent of pragmatic trials, pragmatic clinical trials is to inform decision makers, to enroll a relevant population, and either streamline procedures and data collection or measure a broad range of outcomes. And across 3 domains of recruitment, trial intervention, and measurement, there are various flexibilities that can be introduced that allow for example, broadening eligibility criteria or flexibility in delivery. And these are just examples of questions that you might ask in thinking about how follow-up is being done, or to what extent there's ability for representativeness of patients in the intended use population. And this is an example of, here you see on the far right, an observational study, on the far left, a trial, and the context of how one can be very pragmatic, one can be very explanatory, and where I think we're gonna be is in the middle with thinking about how pragmatic elements can be introduced to allow this hybridization of pragmatic elements, clinical trials, and the meld with real-world data. In other words, ability to conduct trials in routine clinical practice, and hopefully further the learning healthcare system. We're going to skip the polling question. I'm sad that we can't do this polling question because I was going to have, it actually creates a word cloud. But the polling, I was interested in what everybody thinks about the future of RWE. Hopefully we'll get the software resolved and get all that set up. I'd like to just conclude by saying that I think as we move towards pragmatic trials, towards the use of real-world data in drug development, this is really an opportunity for pharmacoepidemiologists. I think that it's such a unique time in drug development, and the ability to think about ways in which the clinical research enterprise can evolve and allow for integration of patient data, collection of data in clinical practice, and reflect that in the real world and actually be captured in a systematic fashion in the context of pragmatic clinical trials is really, I think, exciting, and also most of all exciting because it's patient-centric. It's bringing trials to patients. And with that, it's important that we're collaborative. So I think at the table, having clinicians, statisticians, epidemiologists involved in your study design is paramount to really creating the most robust real-world data studies in oncology. So there are many sources. Studies require careful data selection for data that's fit for purpose. Collaboration, I think, is so important and a strength that we can really bring forward to think about and plan our analyses up front. And regulatory use of real-world data is increasing. So consult our guidances, and the integration of real-world data into clinical evidence can be prospective, patient-centric, and include novel designs such as pragmatic clinical trials. I would like to acknowledge the wonderful team at FDA, our leadership and colleagues across the FDA, specifically two of my colleagues that are here at the conference, Dr. Klutz and Dr. Laro. And I think we're all thinking about how to integrate real world data into the culture of review and really working to advance the science of real world data to generate effective evidence generation for patients. So with that, I would like to just thank everybody so much for your time. The Real World Evidence Program has a website, and we would welcome your ideas and thoughts. And I think that's it. So any questions, feel free to email us. Thank you so much. Question?
While we're waiting, I noticed that a couple of people were taking photos of your slides.
Yeah.
Are you able to make the slides available to everyone?
Of course.
Is it in the system for them to download?
It is.
All right.
Did they upload it already?
Yeah.
Oh, well, that's good. I didn't know about that part, but yeah, everything we have is government property, so we don't. Yeah, go ahead.
I just have a sort of a taxonomy or that we use that term to go around anything from pancreatic protocol, propensity score match, weighted, this compares to the one who actually use which I think epidemiology has been doing for a long time, outside of the health. Do you, the FDA, have a how do you distinguish the various flavors? It's called molecular fusion, I think, in what they consider the spot that they're going to use in different parts that not have different ideas of what it is. At FDA, you guys differentiated the way person. I know you have that sort of different flavors.
Yeah. No, it's an excellent question. And a personal topic of interest to me, I see a lot of external controls. And I have to say, so the FDA does not consider synthetic data as data appropriate for regulatory submissions. So, but synthetic data meaning that it's model data, not based on actual patient observations. I do agree that I think the term synthetic is often not well utilized. But in the context of ECA for how we're thinking it's a direct patient level comparator. And I will say that we have an effort in collaboration with the NCI to work on a glossary that will hopefully address this concern. Feel free to email if there are any others, and thank you.
Okay, thanks everybody. My name is Wei Zhou. In the next about like 20-25 minutes, I'm going to talk about cancer pharmacoepidemiology and pharmacogenomics in oncology drug development. After my presentation, there will be a bio break. So just let you know, you have some time to relax a little bit after my presentation. This is the one actually both for myself and my colleague Nia Sansbury. She is in the middle of changing her job. That's why I did not put her affiliation here. But before I start, maybe give you a little bit of background of myself. So currently I work at a Eli Lilly company, a value evidence outcome research group. I'm leading the oncology group at the VEO. Before that, I worked at like 15 years at Merck in both outcome research and epidemiology. And before that, I did 7 years in the academic. That's where I work with Jeff and not. If you look at his earlier career publications, I was fortunate as kind of co-authors for many of his kind of publications. So this is my disclosure here. My presentation will have two parts. The first one is going to kind of give a little bit of high level kind of overview or introductory regarding pharmacogenomics and targeted therapy in oncology drug development pipeline. Donna made the kind of ask people raise hand. I think about 50% of people here come from industry. So this some of the contact might be not new for you, but I would hope again at least to talk about pharmacogenomics will help you kind of understand better how link the pharmacogenomic information into the drug development. Then the second part is going to kind of give two examples regarding how epidemiologists contribute to the oncology drug development. Particularly, as you know, in recent years, there are many clinical genomics data available right now, many famous names. But there's still sometimes the biomarker may not be readily available into the database itself. So the example I'm going to give here, many talk about the biomarker, may not be readily available in the existing data source. So how, as an epidemiologist, you're going to kind of design your study, answer the research questions to address the need. So first talk about, again, pharmacogenomics and the targeted therapy in the drug development. This is really kind of nice cartoon actually borrowed from Andy, one of his kind of talk before. So patient with the same diagnosis, as we know, is more like a heterogeneous group. Then you can kind of easily divide it. There will be a small group of patients will respond to the treatment, right? There will be some other patients that do not respond to the treatment. And actually there will be some other groups of patients, not just they don't respond to your existing treatment, but they may have adverse kind of event. So the key challenges I think for us to face is to how we can identify the top groups where the people can best respond to the treatment. This is actually a publication a little bit kind of 6 years ago, talking about, again, summary of the data. You can see most of the cancer treatment only benefit a small proportion of the patients. This is not just for particular type of therapies, it's true for chemotherapy, targeted therapy, and immunotherapy. So this is, you can see, I highlighted the kind of column where to show you the immunotherapy in the non-small cell lung cancer patient. You can see even with the kind of pembrolizumab, with nivolumab, and many other kind of immunotherapies, the response rate is only you kind of range it between like 14, 15% to like 40%. So basically, again, only part of the patient respond to the kind of current therapy. And we know carcinogenesis is a multi-step process, right? It involving like genetics, environmental exposures, and also additional kind of factors will kind of become more or less favorable for the development and dissemination of cancer. For when we talk about kind of mutations or biomarkers, right? Like you heard this from Andy's previous talk. We talk about germline, which is kind of more like inherited from our parents. For oncology, cancer patient, most time actually we talk about somatic, which is not inherited, but mainly come from the kind of like tissue samples, either lymph node, cancer kind of biopsy samples. There'll be some other kind of epigenetics. Those are changes unrelated to DNA sequencing, but also may affect the gene expression or phenotypes. I think for most of the targeted therapy for oncology right now, the main focus is on the second part, the somatic mutations, how you can identify the right target to help your drug development. So how are we going to apply the different biomarkers in the oncologies? So you can see when we talk about biomarkers, usually we talk about 3 types of the biomarkers. The most one we talk about called a predictive, which means can you identify a subgroup of patients with a particular biomarker which may predict their response to the treatment. That's what we call the predictive biomarkers. But there will be also many other biomarkers that may not be predictive, may not be for a specific subgroup of patients, but they are prognostic, right? And this will actually help to kind of to see, we call the prognostic factor may not be necessarily related to specific treatment, but it can also affect the outcomes. You can in some way we can see kind of disease stage or even gender, like smoking. Those are kind of sort of the prognostic factor. We know smokers usually have a worse kind of survival. There'll be some other we call kind of pharmacogenomics, like it depends on some of the kind of, particularly in the germline kind of mutations or germline kind of polymorphisms, they may involve in kind of drug metabolism that may change the how big of the dosage will be. So again, when we talk about biomarkers, it's important for us to think about what type of biomarker we talk about. Is it predictive to a particular treatment, or is it prognostic? May not be specific treatment, but also associated with outcomes. Or we actually talk about pharmacogenomics, maybe associated with drug metabolisms or the dosage. This graph I think probably familiar for many of us working in the industry. The top one show you kind of from the drug development from very early targeted discovery, preclinical, until very typical phase 1, phase 2, phase 3, until later. And then the bar here is to show you actually at the different stage of the development from the discovery or drug target identifications until phase 1 to phase 3 kind of drug clinical development. After approval, then what kind of different roles of the biomarker can play. So for example, in the target discovery or the preclinicals, there has been kind of pharmacogenomics biomarkers maybe identify a subgroup of patient may have a kind of respond better or worse as more like a pre-proof of concept studies. And during the drug development, you may also identify whether some patient with a particular biomarker either have a better response to the treatment or kind of have also maybe different toxicity to a specific treatment. And during the development, there are also additional considerations regarding how you want to kind of develop the biomarker to meet with the regulatory requirement. I think Donna touched a little bit kind of requirement regarding what FDA's requirement for the kind of real world data. But here, in order for biomarker to be used as a kind of companion diagnostic to select the patient for treatment, there are theories of a requirement we have to meet with to supporting the development. So this is, again, just one kind of cartoon to show you and the difficulty for the drug development. As you know, the failure rate from discovery, preclinical to phase 1 to phase 2, phase 3 is kind of relatively low. But again, there has been consistent data to showing if the clinical development, you use the pharmacogenomic information to identify the biomarkers, the development success rate will be much, much higher, but also the development cost or the timeline, you can also save a lot. So that's just one way to show you how to include the the genetic evidence will help to kind of not just to increase the success rate, but also help speed up the drug discovery and the development success. This is a not really kind of exhaustive list, but just to show you the different biomarkers has been identified in the oncology kind of development. For example, the first one, BRAF mutations, now has been kind of validated in melanoma, thyroid, colon, and lung cancers. KRAS mutations, as we know, it's being used for select the patient treatment in the colorectal cancers. And in the recent several years, now there are several drugs either already approved or under development for the non-small cell lung cancer. For HER2 expression or amplifications, we know it's a very common biomarker used in breast cancers, but now recently there has been lots of data to show in gastric cancers, HER2 is also very good kind of biomarkers. And I think last year, the ASCO, American Society of Clinical Oncology, there was also very good data to show you some of the antibody drug conjugate can work very well in the called HER2 low or super low kind of populations over there. ALK and EGFR mutations, those are kind of similar kind of biomarkers, just with a different target, has also been used again in non-small cell lung and others. Many of you be aware the BRCA mutations. BRCA mutation actually many times is called germline mutations, can increase the risk of the development of breast cancer or ovarian cancer, but also has been recently, again, with the PARP inhibitors, has been kind of used for patient selections for breast, ovarian, prostate, pancreatic, and others. You may also hear about recently they're called tumor agnostic indications, which means traditionally when doing the drug development or treatment, we try to, based on the classify the cancer by histology. But recently there has been kind of several drugs get approved, which talk about tumor agnostic, which means you do not select the patient based on the tumor histology subtypes. Instead, you select the patient based on the biomarkers. So for example, the microsatellite MSI high, microsatellite instability high, and or mismatch repair deficiency. This has been used as one of the first biomarker approved by FDA as a tumor agnostic. We know now for the RET inhibitors I put here is also use a tumor agnostic. That's one of the breakthrough for the cancer biology and also the drug development where you can target specific biomarker regardless of which tumor type you talk about. So in the next couple minutes, I'm going to give two specific examples regarding, one is talk about how epidemiologists research help the drug target on EGFR alpha mutation in non-small cell lung. The second one is talk about PD-L1 expression in multiple tumor types. Again, both of the examples I'm talking about at the time of the studies, the data set is not readily available for epidemiology to do the research. EGFR alpha right now, as you know, is many very standard biomarker tested. You can, many of the clinical genomic data set use next generation sequence data already cover the EGFR alpha mutations. And the PD-L1 now is also became a more standard testings. So right now, if you talk about those two biomarkers, many of the data set may already available to do the research. But the example here is actually in the much earlier development. By that time, the biomarker itself is not available in the data set. So therefore, epidemiologists in collaboration with others has designed the epidemiology studies to help with the development. So some of you may know gefitinib is one of the first EGFR inhibitors. Again, it's target on EGFR alpha kinase inhibitors. It was received accelerated approval from FDA in 2003, that's 20 years ago. And the approval was based on, again, single arm trials where treatment of patients who have failed 2 or more courses of chemotherapy. So which means there's a huge unmet medical need over there because after a patient that time failed the chemotherapy, there's no options. And also historically, the response rate was very low, and in the early trials, the objective response rate is about 10% for gefitinib. As you know, in the US FDA, even you get accelerated approval, you have to continue to do additional randomized phase 3 trials, use the overall survival at the endpoint before you can convert into the traditional approval. And so in 2004 December, their phase 3 trials actually failed because it did not show any survival benefit over comparison with the placebos. And because of that, FDA basically limited the use of gefitinib to only among the patients who already currently receive treatment and are benefiting from the treatment. And their planned submission to EMA was withdrawn. But also during the trials, they did some additional subgroup analysis. They found actually among the patients from Asian origins and non-smokers, and among those subgroup analysis do find a better survival among those. So the question is that, is response to gefitinib is predictable? And also it's very interesting, same year, 2004, there are 2 simultaneous publications almost come out same time. One come from Mass General Hospital, another one from the Dana-Farber Cancer Institute. Almost same time, one published in New England Journal of Medicine, another one published in Science. Both of the study actually showing that if patients have a somatic mutations in the EGFR alpha exon 18 to 21s, and they can predict the response to the gefitinib treatment. So I think that the next steps, the company did not just the company, but also academically did some additional work, epidemiology work to show actually how you can enrich the patient populations who may have a much higher EGFR alpha mutation frequencies. So this is one of the review articles kind of published like 2 years later. You can see here, both of the slides are showing, again, the left one is showing among all non-small cell lung cancer patients, adenocarcinoma, liver smokers, Asian, and females have a much higher prevalence of EGFR mutations when you compare with squamous cell carcinoma, with smokers, with Caucasian population or non-Asian populations, and also males. The right one is show you again, in the adenocarcinoma patient, you see the same things. Liver smokers, Asian, female has much higher kind of prevalence data. So those are additional epidemiology data show the EGFR mutation frequency is actually different. So I think the company did a very, very smart studies. They did additional phase 3 trials. They called IPAS studies, open label, randomized study, again to assess gefitinib with chemotherapy in the first line setting. The primary endpoint was progression-free survival. Then you enrolled more than 1,200 patients from Asia, and also tumors, again, many in the adenocarcinoma, and also liver smokers and the former light smokers. And during the trial, actually, I think one of the smart things the company did is AstraZeneca is the company developed this one, is that they understand for metastatic kind of non-small cell lung cancer patients, sometimes it's very difficult to get the tissue samples doing the biomarker testing. So that's why instead of to force every patient, you have to have EGFR mutations before you enroll into the trial. Based on the epidemiology data, they try to enrich the trial patients, where again, in the Asian, in the adenocarcinoma, in the female and liver smokers. So that's why in the end, again, the trial was very successful, and the drug, again, now is approved in metastatic non-small cell lung cancer patients with active EGFR mutations. Again, this is the first generation of the EGFR inhibitors. Now we are talking about second or third generations of this. This is actually a very, very good example to show how epidemiology studies can help identify the determinant of the frequency as well as the distributions of the associated biomarkers. I think particularly when colleagues you work in the industry, sometimes your clinical teams may have interest in to use this biomarker for patient selections. I think it's your job to say how we can do additional epidemiology studies to understand whether any patient characteristics might be helpful you to enrich the population. You can use histology, race, ethnicity, age, or some other factors. And the second one actually is the studies I've been kind of personally kind of leading the epidemiology studies, talking about PD-L1 in non-small cell lung. So this is again graph to show how the PD-1 kind of inhibitors here is talk about pembrolizumab, biologically how it work into the kind of the cancer development. Again, PD-1 is a immune checkpoint receptors. They have two kind of receptors, the PD-L1 and the PD-L2 binding together. That's where kind of have the tumor to increase. You can see there actually among PD-1 and PD-L2, PD-L1 probably play kind of much more important roles over there. That's why in the immunotherapy, you see there will be drug target on both PD-L1, PD-L2, but there are also several drugs only focused on PD-L1. And again, PD-1 blockade through the therapies will reverse the bindings. That's how the drug works. PD-1 expression actually was among the earliest biomarker evaluate can be predictive to the anti-PD-1 treatment. Actually, in the first publication from BMS in New England Journal of Medicine 2010, that time there also phase 1 data that also showed again patients with PD-1 expression, higher expression, kind of respond better to the kind of treatment. But again, PD-L1 is measured by its protein expression. So it's measured by immunohistochemistry method. And at that time, there is no readily available kind of data in the data set. And also the challenge is also, again, there has been different antibodies, different methodology, and different cutoff to define what is positive versus negatives. So therefore, you look at the literature, there might be some publications over there, but their prevalence is everywhere from 10% to 90%. Just talk about non-small cell lung cancer patient. Again, it's because they use a different antibody, different method, and different cutoff. And also there has been very kind of not consistent result on whether PD-1 kind of expression is a positive or negative prognostic kind of factor. And why this question is very important, because in the early development of pembrolizumab, it was single-arm trials. We were trying to see whether patients with high PD-1 expressions will respond better to the treatment. So this is the predictive biomarkers. But because of the single-arm trial setting with no randomizations, it's also very important to understand the prognostic effect of the biomarkers. You want to show whether the patient better survival or better response to the treatment is because of the drug itself, or is just because it's positive prognostic factor. Doesn't matter what kind of treatment to receive. So that, again, understand the prognostic effect is important in every setting, but particularly important in the single-arm trial settings. So well-designed epidemiology study, again, use representative samples and valid technologies are needed to address these questions. So we want to design a study to use the same antibody, same method, and same cutoff as we use in the clinical trials. So the scientific question is actually, because that's where Donna told us, everything is starting from the research questions. So the research question here we have is, what is the PD-1 prevalence among non-small cell lung cancer patients? And second is whether PD-1 expression is the prognostic factors among patients that receive standard of care. The challenge again, is that time there was no existing data source available. And we basically have to identify investigators who have collected patient non-small cell lung tissue samples, and also who we can access kind of to the clinical informations. And also, we also kind of have to work with investigators who actually can ship those samples to the central lab, because we want to use the same methodology, same cutoff as the clinical trials. So those are the three kind of challenges which are kind of facing in order to answer the questions. In the end, we did two studies, one kind of among Caucasian population, another one from Asian populations, because we understand there is a huge kind of ethnicity difference of the prognosis in the non-small cell lung setting. So those are the results of the two epidemiology study. The left one is a study we did in Denmark in the metastatic setting. The right one is the one we did in Korea. Again, most of them are patients who receive surgical samples. So therefore, many of them are kind of more like early stage patients. That's where you can see the survival curve are really kind of different. But the result actually kind of relatively consistent of the both of studies. Our result again use the same assay, same cutoff as using in the clinical trials. We found again PD-1 expression is not a positive prognostic factors, which means what we see in our clinical trials, it's a positive predictive of the biomarker related to treatment. So what are the benefits and challenges for pharmacogenomics? We know it can advance, again, risk stratification for the disease. It will help, again, the drug discovery and development process, identify a specific target. Again, earlier can be with the need identifications, optimizations. The example actually give like gefitinib is actually very good examples where previously failed drug candidates can be rescued in some ways. Again, when you're trying to match with the niche populations with the particular biomarkers. Of course, the third one is also can help speed up the development as well as approval timelines. So this is my last slide. Again, in summary, there has been very good progress in the oncology drug development in recent years, particularly in the targeted therapy and immunotherapies. Again, pharmacogenomics and the biomarkers play very central roles in the targeted therapies. I hope I give you kind of at least some of the primer for you to think about. Again, epidemiology play very, very kind of central roles in the oncology drug development, where we can either analyze the existing data source or maybe collaborate with the investigators to generate the data which may not be readily available. Of course, everything here is to collaborate with cross-functional group, clinical biomarker stats, and others. So I'll stop here and to see whether any of you have any questions before we take the break. Thank you.
Dami Onasaya from Carolyn Research and Bishop Maryland, Baltimore. You just reiterate how identifying biomarkers speeds up the timeline. Just a little more on that.
Yeah. Well, first, your drug may only work in the small subgroup of patients. By identifying a biomarker which may be predictive of that treatment, which means you can see much larger effective size than you in the overall populations, because it get diluted in some way. So therefore, you can design your trial with a much smaller sample size because you see much larger kind of effect. And also enrollment will be much quicker because your sample size is smaller, right? And also sometimes, again, depends on the context, whether there's a huge unmet medical need or not. You may be able to use a single-arm trial settings to get accelerated approval as the first steps. So there are several reasons will help you to, again, not just increase the success rate of your development, but also help speed up the timeline and also save the cost. Yeah, you can compare. There is a huge difference between like this 200 or 300 patient trial versus you have to do like 500, 600, 800. Yeah. Any other questions? If Andy, how much time we have for the bio break? Or? So come back at like 4 o'clock. Okay. So yeah, take a break, get some coffees, have a chat, and then next will be Bruce and Jeff. Their talk will be much more exciting than my talk. Okay, just for your information. Thank you.
All right, yes. Set it up. So.
Okay, welcome everyone back. So I'm Bruce Carlton, and I am at the I'm the division chief and professor in the Department of Pediatrics at the University of British Columbia, which is actually further away from Halifax than Europe. Gives you some idea, if you don't know the geography of Canada, how far we are away. So I have a number of government sources that fund the work you're going to see today. They're all federal or provincial agencies, and I've done some consulting with a couple of companies as noted on this slide in this particular area. So what I want to do is take you through the design of a pharmacogenomic study and basically 20 years of thinking about a given adverse event and what I've learned from this and why pharmacoepidemiology is really important in this area and why we need you to help with this. And so when you're designing pharmacogenomic studies, the things that I tend to think about, the things that we tend to do is we think about what are the candidate genes that we might find are relevant. And so the first thing is, what are the genes involved in biotransformation, absorption, distribution, metabolism, and excretion? What about the genes involved in the target, the receptor, so pharmacodynamic targets? And what is the literature found as relevant or non-relevant? That's the first step, is I begin parsing these together. And I always do that even though there are genomic arrays that will cover these things. I want to know in advance what I think I might find as as relevant candidates. The next step really is a genome-wide scan, often called genome-wide association studies. And then I can look to see whether the candidate genes that I've identified in the earlier analysis are on the panel that I'm using, and if all relevant variants are there. So my friend who owns a car dealership, years ago when I bought a 5.4 liter V8 Ford F-150 off-road, he said gets 45 miles per gallon. Of course, your actual mileage may vary, which is the golden asterisk, right, on sales. And the same thing happens with these companies that produce arrays for genotyping. They'll say covers CYP2D6, which is a very common cytochrome enzyme involved in drug biotransformation, but in fact, they don't cover all the relevant variants. CYP2D6 is highly polymorphic. There are probably 100 plus known variants. There are many, not 100, but there are many that are relevant variants. And most of the commercial arrays do a poor job that Illumina produces at covering that gene in its entirety for drug-specific or pharmacogenomic studies. So it's always important not just to know what gene they cover, but what variants they have within it. We can always do whole exomes, or what actually we don't call it whole exomes anymore, we call it exome sequencing because it's not really the whole exome, or we could do whole genome. And the reason that I avoid doing those things initially is cost. You can see the price goes from $50 all the way up to $5,000. I saw a slide today suggesting that whole genome was $500. I don't know where they're getting that. That's not what the cost is, and for whole genome. And more data, as you know, as epidemiologists, does not necessarily mean better outcomes. It means you have more to work through, more multiple testing errors that are possible. And so I love the sequential step approach of thinking about the design of a pharmacogenomic study. Now, before we think about the genomics, we have to think about what the adverse event in the case, that's what I'm going to talk about today, is, and how to find those and what the phenotype really is. So this is genotype, but phenotyping is often found through rapid cycle analysis or active surveillance. Rapid cycle analysis is used in vaccine studies. We use it a lot with studies I'm now doing with the US Centers for Disease Control with COVID vaccines, in which you look for outcome rates for certain types of reactions. We're looking for pericarditis, myocarditis, myopericarditis, Guillain-Barré syndrome, and vaccine-induced thrombotic thrombocytopenia in COVID-19 vaccine recipients. And we do that, we look both within the risk window and outside the risk window and to compare. So we would expect people to develop Guillain-Barré syndrome within 42 days of vaccination, but people develop Guillain-Barré syndrome for other reasons, like COVID infection, outside of that interval, and we would compare these intervals. And so this is really important in drug analysis as well, to sort of be thinking about the phenotype, thinking about the reaction itself, and whether you're comfortable with the fact that this is drug-induced or caused by something else. In many cases, concomitant medications are also involved or used in a patient that are also potentially implicated in an adverse event. So it's important to get a clean understanding of what you're looking at. We do a lot of active surveillance to get that to clean that up, and we generally use clinicians that we've trained in adverse drug reaction recognition, evaluation, reporting. We work with specific case report forms. They're employees of the network, so they're not doing this off the side of their desk when they have other things to do. This is their job. And it's not, and the aim is to have full reporting, but not every adverse event, just the ones that we're actually looking for. And the aim is, of course, to have as much critical data as possible. And this is easier to work when we work with employees. We can standardize and develop thorough data collection processes and critique their quality through a quality assurance process, what they're doing. So we make sure we have complete data. We now have 32 sites in Canada. that do this work, and it's been very successful. We have about 120,000 adverse drug reaction cases and drug exposed controls over the last 20 years from working on this. So that's a little background. Now I'm going to show you a drug and show you some of the output that we've done. So cisplatin is used as a drug of choice for solid tumors in pediatrics. We don't have a lot of solid tumors in pediatric cancer, but there are some. And about 1 million patients receive cisplatin every year in North America and Europe. This is a drug, because it is a heavy metal, it is platinum based, causes permanent hearing loss. And that varies across the age spectrum between 10 and 38% of patients overall, although there is increased frequency and severity in children. Somewhere between 30 and 60% of children develop some level of severe hearing loss. And I'll explain severe in a moment. And more of those children who are younger develop severe hearing loss. So there seems to be an age or a concentration dependency here. And I can tell you that at the British Columbia Children's Hospital, almost 40% of our patients have developed CTCAE, common terminology for criteria of adverse events, grade 3 or 4 hearing loss since 2005. That would be the need for hearing aids or cochlear implants. This is very standard at most institutions. We all use the same protocols for treatment, so I wouldn't expect that to be much different between the centers. And so I thought I'd start with 2 cases. These are children that are essentially the same age, the same diagnosis, the same longitudinal time course of diagnosis, receive the same chemotherapy, and both of these children are alive and well today as a result of cancer chemotherapy that was successful. In pediatric cancer, the 5-year survival rate really defines cure. If you don't have relapse within 5 years, you almost never will. So we define that as cure. So these kids are both cured as a result, partially for their cisplatin and other chemotherapy and treatments they received. But although the cases sound familiar from the same tumor, the same protocol, the same good outcome point of view, there were significant differences in their audiograms. And the second case needed the last 2 doses of cisplatin held due to significant hearing loss, and that case needed and had received permanent hearing aids, will need hearing aids the rest of that patient's life as a result of this toxicity. Now, note that the last 2 doses were held. So this child received less chemotherapy than the other child. Both are alive and well today. So there's something interesting about that we reduce the amount of cisplatin exposure, and yet the child still survives. So this is a big question that I have when I go into these studies is, are we overtreating some children, and why might that be? So to familiarize you with an audiogram, in case you're not, the normal range in pitch and loudness, in other words, in decibels and hertz, for a child and an adult are listed, are provided in the gray zone. Children actually have a little bit more high frequency hearing than adults, that they lose over time. Years ago, the convenience store chain 7-Eleven used to put this high-pitched noise device outside the front doors that made it like an irritating sound that only kids could hear, adolescent kids that tend to loiter and hang outside of those stores, and that would drive them away, but people like me would never hear it, and so wouldn't be bothered by it. But I was never hanging out at 7-Eleven in my adult years. But this is normal range. So you see the right and left ear should be in that gray zone across the pitches. And then as you develop hearing loss, your curves decrease. And when you get even to moderate hearing loss, you lose the soft consonant sounds. So if I were to say to you, I'm really looking forward to going home on Unday, that's what you heard, you would understand, because you understand language, you would understand I meant Sunday, and your brain would just fill in that missing sound, and we would continue our conversation. But when you're a child and you're developing hearing, you don't have that contextual knowledge of language. And so it becomes much more difficult for them socially to develop when they can't hear soft consonant sounds. And there are lots of words with soft consonants. And so that's why even moderate hearing loss is considered severe. So here are audiograms at baseline for both of these children, showing normal right and left ears at baseline. Halfway through cisplatin, midpoint of therapy, you can see that one child has significantly reduced hearing in both ears, one more than the other, but both ears are affected and the other still remains normal. And then as we look at current studies, we can see a progressive worsening even of that case beyond now. So cisplatin is a drug which is a heavy metal. Like lead, it is in the body for a long time. You can measure it in the plasma 10 years after treatment. In many cases, even 20 years after treatment, you can still find cisplatin. So it is not an adverse effect like others where if you, oh my gosh, you know, their renal function is declining, stop the aminoglycoside, it's too late. These are long-term resident drugs, and that's why we're concerned about them. So why does one child get hearing loss and another not? So what we began doing is taking 162 children. Again, these are rare tumors. So even across a country like Canada, it took us quite a while to accrue this number of patients. And we ended up with 106 cases and actually fewer patients with normal hearing because the frequency of hearing loss from cisplatin is greater than 60% in children overall. And so we ended up with a small number of controls. And the cases were defined as a level of hearing loss that would be what we would intervene with. So if we're looking for genomic biomarkers of risk, I'm going to use cisplatin. I'm going to treat this kid's brain tumor. I'm not going to not use it. So I really don't care so much about mild hearing loss. But I would like to know who I'm going to have to monitor more frequently or who I'm going to have to intervene with hearing aids, et cetera. And so that became the criteria. This confuses all of my graduate students to no end who can't stand the fact that we end up throwing out patients who end up with grade 1 hearing loss, and they want to include them. And so they send me analyses, even though I tell them not to do this, saying, I've included the grade 1s. Why? I don't really want to know if somebody has mild hearing loss, partly because as a heavy metal, this hearing loss will continue. So I don't know if it's a transition state. They're grade 1 now, but they're going to be grade 2 later, or they're going to stay as grade 1s. I'm not sure. And so then they send me back the analysis and say, I lumped the grade 1s with the grade 0. Now you've contaminated your control group with somebody who has hearing loss. So I don't like that either. Just leave them out. But we'll lose power if we take these patients out. Yes, we will, but I want a clean analysis. So I always say, focus on the extremes. Focus on patients who have a clinically relevant level of an adverse outcome versus those that don't. And let's find a biomarker that can educate clinicians about risk in those populations. And from this work now published in 2009 in Nature Genetics, we found 2 particular genes were involved in hearing loss. Really surprised us. The methyltransferases. Now, methyltransferases are traditionally associated with the disposition of thiopurines. That's why they were on our panel. But they aren't necessarily associated with cisplatin-induced hearing loss or cisplatin biotransformation. But if you know anything about homocysteine and reactive oxygen species pathways, the methyltransferases are key components, and they're not just involved in thiopurines, they're involved in the generation of reactive oxygen species, et cetera. They're very important genes. And from this work, we found very significant odds ratios showing that these were risk variants of significance in patients with hearing loss versus those that got cisplatin exposed that did not have that. And we posited that the cisplatin was binding purines. It was causing cross-linking and cell death, and TPMT normally inactivates purine compounds like cisplatin. We thought that COMT was probably, and TPMT were using the S-adenosyl L-methionine substrate. You accumulate this substrate and it's toxic in the presence of cisplatin. That was our sort of posited work in 2009 in Nature Genetics. And we looked at combining the top 2 SNPs in both genes and could show that we could identify about half of the cases of ototoxicity with these markers, which isn't that great, but we could do it with a very high specificity of about 93%. So if you had the variants, you were going to go deaf, or at least you were going to need hearing aids. And if you, but that didn't mean that if you didn't have the variants, you wouldn't have hearing loss from some other cause. And so this was not all of the story, but it was part of our early story. And we published that, and we showed that if you increase the number of risk alleles that a given patient has, there's an increased severity, an increased frequency, and an earlier onset of hearing loss. FDA reviewed our data on its own. We sent them the raw data. We were able to do that at the time with permission from the REB, and resulted in a label change that came and went and came back in 2012, including information about thiopurine methyltransferase gene and the risk of cisplatin-induced ototoxicity being increased. So this is interesting. They didn't accept the COMT results because they felt that the population was too small, which is fine. And then we began the process of replicating this study, thinking, let's look at an independent cohort of patients to see what we find. Well, guess what? We find different things. We found TPMT show up again, but we found ABCC3 as a genetic variant that was highly associated with cisplatin-induced hearing loss. And we published in this in CPT, we published this Kaplan-Meier curve showing that you could differentiate patients into high, intermediate, low risk of hearing loss, depending upon the 3 groups. I'm sorry that the COMT was still included in this variant, but it was less significant. And so this was confusing to us because we were looking to have exactly the same results, not find new variants and find less relevance of COMT only when it was included within the group. And so this is what I call the risks of the drive to replicate. That replication becomes the idea that, well, that study was published, but hasn't been replicated. Well, you know what? You can replicate a poor study. That doesn't make it a better study. And really, published methods are an abstract of the scientific process. So I would say that our Nature Genetics paper in 2009 was 5 years of hard work of about 12 people. It involved a massive amount of work, and we were given less than 3,000 words to report that. So if you read that study and thought you could replicate based upon that alone without talking to me, you're sadly mistaken. And I think all of us as scientists that publish recognize that this is a problem, that reading a study and trying to replicate it from what is really an abstract of years of work is inappropriate. And there's little way for us to share the fulsome nature of the scientific process, unless you're my postdocs and my graduate students who think that the supplemental material is the place to do it. My God, I can't believe these papers I get sent from them with 49 supplemental figures and text, and they think putting everything out there is somehow going to work, but it's not organized in any way. It's just stuff. And so I don't think that's really the way to do it. I think we really need to find a way to truly understand the multidisciplinary nature of pharmacogenomic risk studies in cancer and work together in each other's labs to understand what we're doing so that we can actually truly replicate our work. And as I was thinking about this, the National Academy of Sciences published an editorial looking at the issue of climate change. And unless you've been hiding under a rock, the debates about climate change, if it's real or it's not, in some people's minds is a big debate. And they'll point out that this study shows that there's an effect of climate and this study doesn't. So what are we supposed to believe? And so in the proceedings of the National Academy of Sciences, they put this editorial out, and it's a lot of text here. I'm just going to highlight a couple of things. Don't worry about reading it. My slides are available on the website, so you can get them. But essentially what they're suggesting here is that reproducibility or replication is neither a necessary nor a sufficient condition to ensure the validity of conclusions drawn from the research. As I mentioned earlier, you can replicate a bad study. That doesn't make it a better study. And certainly reproducibility and replicability play an important role in achieving rigor and transparency. And for some lines of scientific inquiry, replication is one way to gain confidence in the scientific knowledge. It doesn't mean throw out the idea of replication, but understand its limitations. And this last sentence is the thing I think is most important in their report. The robustness of science is less well represented by the replications between two individual studies or reproduction of one or more studies than by a more holistic web of knowledge reinforced through multiple lines of examination and inquiry. I can't tell you the number of times clinicians look at epidemiological studies and say, observational. I mean, there's no causal inference I can draw between these two conclusions. Well, okay, but don't throw it out yet. What about looking at a pharmacogenomic study that attempts to take a signal that a pharmacoepidemiology group has developed and replicates that using a different method through active surveillance, for example, or rapid cycle analysis, finds the same finding? That strengthens the case that there is an adverse event of concern and helps us to understand more of the biology underlying it. And so when I was working through this process and people were criticizing our work for TPMT, how can a thiopurine methyltransferase be related? Oh, you must have made some mistake in your analysis. You begin to look at other studies that are published, some of which I had reviewed officially, and I began looking at the differences between them. So numbers of patients are one, obviously, criteria that's important to look at. How many cases, how many controls? Small studies can be problematic or informative, both cases. What grades of toxicity for cases and controls were used? You can see that they differ between studies here. What grading scheme was used and how do they relate to each other? Also important, we published on that. What age of the patients exists? Epigenetic findings and ontogeny in pediatrics means that we're dealing from birth to adulthood. 0 to 18 is kids, but let's face facts. A neonate is not an adolescent, is not an adult. They're all different. Tumor types, how does that influence? How about concomitant radiation, right? So, which to the head means you're hitting the ear, right? So the failure to detect significant associations may be the fact that this heterogeneity between populations in these rare tumor types is too significant. So in the case of the Yang study, all of their patients got cranial spinal irradiation. In the case of my study, none got cranial spinal radiation. There's a significant difference. Over 90% of the Yang patients received amifostine as an otoprotectant. None of our patients received amifostine. In the Landvers Comiskey study, they had shorter follow-up times. They didn't account for confounding factors like concomitant medication use, cranial irradiation, concomitant otoprotectant use, length of follow-up, ancestry. So all of these studies have problems, ours included, but they're different. And the abstract nature of the way we publish is a problem in trying to replicate. That's my point. And then I began looking at this issue that Dr. Friedman mentioned to begin with, thinking about cumulative dose. Well, if you're looking at anthracyclines and cardiotoxicity, cumulative exposure makes complete sense because you don't see cardiotoxicity until the end of treatment. There is a cumulative effect of anthracyclines on causing cardiotoxicity. And we know that because we actually follow cumulative exposure and dose limit patients because we know the probability of toxicity increases as cumulative exposure goes beyond certain thresholds. But in the case of cisplatin, here's two groups of patients, cases and controls. This is now later work that we've published more recently, I think last year. It might have been published earlier this year, where we showed that the cumulative exposure between cases and controls is exactly the same. But if you look at the dose to toxicity, it's significantly different and it's lower in cases because the toxicity doesn't occur at the end of treatment, it occurs within 4 to 8 months of beginning cisplatin therapy, right? You're on therapy, you're on cisplatin, and then you're off, and then you're on the next cycle, and you're off. You're on at the next cycle, you're off. And so the amount of cisplatin they receive before we detect hearing loss is not it's not at the end of treatment. This is why we want serial monitoring and not just, well, we'll find out what's going on at the end. The International Harmonization Guidelines that I'm a co-author on, despite the fact that I disagreed with this and fought for this, is to actually have one audiogram at baseline and one at the end of treatment. It's not to do serial audiograms. And that's because the hepatoblastoma folks, who most of those patients whom have hearing loss, don't want to be having serial audiograms and having to tell parents that their child is going deaf and getting these audiograms and showing that as they're progressing. They want to continue therapy, get done with it, and then deal with the hearing aids, which is an interesting idea. And I understand the reason for that, but clearly there's a dose to toxicity difference between case and control. So when I read papers still today that say high cumulative exposure cisplatin leads to hearing loss, the answer is no, you're wrong, it does not. So we began thinking about this further, and I said to one of my students at the time, why don't we group these in terms of a time variable? Let's add time into our cumulative exposure. And so what about dose intensity? So how much cisplatin are they getting, and in what level of intensity? So if you look at these different boxes that I have here, the last line on every, which you can't read, but you will in the slides that you have available to you, you'll see that the cumulative exposures are fairly consistent between different regimens. But the way we've described these in terms of intensity is if you get the dose, let's say 90 to 100 milligrams per meter squared over one day, that's high intensity. If you get 20 to 30 milligrams per meter squared over 3 to 5 days, that's low intensity. Same amount of cisplatin cumulatively, same dose, but one's day one and one is over 5 days. Very different, 3 to 5 days. And so and then you have intermediate intensity in between those two extremes. And so we plotted the cumulative incidence over time between these groups and found very significant differences. The moderate and the high intensity protocols result in much greater levels of ototoxicity than the lower intensity groups, and they tend to plateau later in terms of months. You can see that the plateau happens about 6 months for low intensity patients and about 8 months for high intensity or moderate to high intensity patients. So this is a very interesting finding. And so my first question to the head of COG was, who's Doug Hawkins at Seattle Children's, not far from us in Vancouver. I said, is there evidence that the high intensity protocols need to be given? Cisplatin needs to be delivered over one day in order for survival to be achieved. In other words, we can't go to a 3 to 5 day regimen because their survival will be compromised. And he said, no, there's no evidence for this, it's just tradition. That's interesting. So you can see between 25 and almost 70% hearing loss, significant difference just based upon the intensity. Now, when you add in the genetic variants, you see something else really interesting. You see the same orange and green lines, but you also see that patients, or red and green lines, but you also see that having these risk alleles, we don't have very many patients. I put them on the bottom, so the numbers are small, but you can see that if you're on a low intensity regimen and you have a genetic factor that increases your risk, the increased risk is small compared to if you have a high intensity regimen and then you have a risk allele. And so this is another factor in genetic studies, which is to begin examining in what group of patients the genetic markers are most critical to deal with. And so this was really a shift in our paradigm shift in our thinking about what we ought to do next. And so maybe we ought to pause now and think a little bit about using cisplatin in lower intensity environments or making those comparisons. Now, trying to convince well-meaning oncologists, and I know many of them, they're all well-meaning, to change the intensity of the treatment in hopes that it doesn't change survival is a very uncomfortable concept. And so this is something that we're currently exploring. But going beyond that, again, triangulating evidence from different sources, we began exploring why TPMT would affect cisplatin. And so the first thing we did was we expressed these variants in two mouse inner ear cell lines, the house ear instituted organ of Corti 1 cell line and the UBOC1 cell lines, and monitor the impact of these TPMT variants on cisplatin response, looking at cell death and also activation of a cisplatin response gene that we found after a candidate work in a genome-wide study called TLR4 or toll-like receptor 4. And what we found is that the STAR3A variants definitely sensitized the cells to cisplatin cytotoxicity compared to wild type. There were significant differences in the IC50 concentration in both cell lines in the patients that had these low activity TPMT variants. So they have this cellular phenotype consistent with essentially higher effective cisplatin concentration. So maybe these variant carriers need less cisplatin than the others because they have effectively higher concentrations. That's one idea, at least in the middle ear. The next step was to look at using a TLR4 induction as a cisplatin biosensor to look at whether the expression is induced by increasing cisplatin concentrations. And in fact, it is. Toll-like receptor 4 is a particularly important inflammatory biomarker. And we showed that as you increase the concentration of cisplatin in these cell lines, we find that TLR4 induction is also increased. And then we looked specifically at the wild type cells, the STAR1 cells on the right at the bottom graph, and the low activity variant cells. And we showed that these low activity variant, the STAR3A cells, had significantly increased TLR4 response to cisplatin, which is basically they have more inflammatory response, and that's consistent with higher effective cisplatin concentrations in these particular cells. So moving forward, we've studied TLR4 a bit more. One of my postdocs at the time has now moved on to a faculty job, and he published this work looking at deleting TLR4 to see if we could actually decrease cisplatin-induced cell death. And in fact, we can. If we delete TLR4, we can increase the viability and decrease cell apoptosis, looking at two different methods to do that. And so that began our focus of how TLR4 mediates cisplatin toxicity and what drugs can we use to do this. And so if you delete TLR4, you decrease the reactive oxygen species generation that occurs in this homocysteine ROS pathway, and you also decrease the pro-inflammatory cytokine cascade that occurs in response to cisplatin. So we searched for small molecules using some computer-aided drug design work that we've been doing to find compounds that are commercially available that we could use to see if we could prevent cisplatin ototoxicity. And this is the result of that work, showing that TAC242, a Takeda compound, also known commercially as Restoravid, can actually reduce as a TLR4 inhibitor, bind to the intracellular TIR domain of TLR4 and disrupts adaptor protein recruitment, allowing for less cell death and less cell toxicity. We've also looked at rapidly dividing cells to see if using a TLR4 inhibitor would also impact cancer cell death, and it doesn't. So this provides us some early evidence that we may be able to deal with this particular problem with existing drugs. There are lots of TLR4 inhibitors out there. Restoravid is one. Hydroxychloroquine, President Trump's favorite drug, is another TLR4 inhibitor. And which ones might be able to be used in this context and studied, not used clinically, but studied as their potential impact, it would be interesting. You'll notice also in this graph, I just wanted to point out on the left side that we looked at lipopolysaccharide and nickel and cisplatin. Nickel is a known toxic compound to humans. For those that have babies, if you have a child with nickel allergy, they'll often have the snaps on their little clothing, and they'll develop little target lesions right around the snaps, a skin reaction to nickel. It's actually quite common in children, and we wanted to see if this was a particular metal problem, and we saw it with nickel as well. So this is a metal-related antagonist. So I began thinking, well, could we use a TLR4 inhibitor like like Restoravid in patients? And the answer is not yet. I think we need more information to do that. And this is what I think is data that's sufficient to act on pharmacogenomic findings. You need to have it in the drug's label that there's an association that is strong and consistent. There are guidelines published about pharmacogenomic information by expert consortia that are peer reviewed. That's another source of that. I particularly am interested in clinically actionable findings that are demonstrated and replicated in at least three worldwide independent populations that are as similar as possible. And I try to strive for an effect size of at least 3 or higher. And I figure since cholesterol and heart disease are linked, high cholesterol and heart disease are linked by an odds ratio of around 2. This is a magnitude higher and more impressive than that. And yet we spend a lot of money on statins for an odds ratio that's kind of at the margin of clinical relevance. But most important, I think, we want to understand the mechanism by which this cisplatin ototoxicity is occurring first before we start using this in practice. But I think through this process of nearly 20 years now, we're beginning to understand that maybe it's the intensity of cisplatin that's important here in some patients, and genetics are important as well. And so we continue to do this particular work. My take-home messages for today are the 3. One is there's no valid genomic marker without rigorous consideration of the phenotype. That's why pharmacoepidemiologists are critical in our work and always will be. Simply looking at the numbers of studies with similar pharmacogenomic findings is insufficient to identify clinically relevant findings. I make this point because I continue to review papers that say 3 studies found an association and 3 didn't. Well, first of all, and I know that the word association could mean what? Weak association, strong association. I don't even know what to make of that. And numbers of studies does not portend the rigor of the data. So if I'm publishing in Nature Genetics, which is one of the arguably top 4 biomedical journals in the world, the requirement for independent replication is higher than the British Columbia Medical Journal, where I could publish a case report. They're both studies, but they should not be compared. So I don't like it when people compare numbers of studies. And lastly, genomic information is really important, less about predicting in whom the adverse event is occurring, but it informs the mechanism of drug-induced harm. What's the mechanism? That's how we're going to solve these problems and come up with solutions to drug-induced toxicities, is by understanding the mechanism, the biology that underlies harm. Thank you very much.
And I'm curious about in the case of beginning 2 children with the same treatment strategy and one with side effect and the other without, how do you rule out the environment factor and lead a genetics study?
How do I rule out an environmental factor as the cause before I start the genetic study? Right, good question. And so the answer is, and this is one of the wonders of studying rare diseases like pediatric cancer, where we have few cases, is we can do a baseline assessment of their hearing right before we begin cisplatin. Okay, so we know that these children had normal hearing before they began cisplatin, and abnormal hearing developed afterwards. That's harder to do in adult populations because the number of cancer patients is much higher. And so you have to have adequate resources to, let's say, monitor the heart, to do routine echocardiography, or to do audiology if you're dealing with large numbers of patients. In pediatric cancer, because it's a rare condition, in the United States, about 15,000 cases a year, which is fairly small with a population of 380 million people, almost 400 million people. It's easier to organize resources to do that type of monitoring. Does that make sense? So that's how we ruled that out. But it's an important question. The other issue I wanted to point out is that any case of unilateral hearing loss where one side is affected and the other isn't, that's not cisplatin. So we've ruled that out. That's more likely to be noise or exposure-related hearing loss. People that like to shoot guns, target shooting and so forth, can have affected. hearing and so forth, but it has to be bilateral for it to be cisplatin. And almost all kids, when they do have hearing loss, it is bilateral.
Before the study, everyone doesn't know that it's related with the genetic, right? But how I mean, how do you think that it may be the genetic factor make the ADR?
Right. So the question is, they're both normal hearing to begin with. We've done a normal baseline. Both the cases and the controls are normal. So these are all cisplatin exposed patients. And then what we do is we study the genetics of both populations divided as like a nested case control or a case control study. We divide them into those children with hearing loss and those without. And you'll notice that we had fewer controls. We had fewer patients without hearing loss, but we find the differences in the genes between those two populations. You're welcome.
Thank you for a very interesting presentation, Dami and Osaya from Carlon Research and University of Maryland, Baltimore. I'm asking this question from the perspective of a trainee in pharmacoepidemiology who has never seen or handled a pharmacogenomics data set, or will probably never enter a pharmacogenomics lab to use. Well, maybe
We can solve that problem.
Yeah. So what's your recommendation, advice to primary pharmacoepidemiologists who think there's an association between a drug and an outcome, and where there might be other literature suggesting some genetic or phenotype influences to that association? What can we do differently? Is this something, in my mind, it's on the spectrum between, okay, your discussion section, you can say maybe part of why we didn't find a finding is because we didn't account for these pharmacogenomic subtypes. Or maybe at the other end of the spectrum, perhaps there's data somewhere where we have the things we use in pharmacoepi, i.e. drug utilization, day supply, and then there's information on genetic biomarkers. How can we do our research differently, I guess?
Yeah, I mean, it's a really great question. And the answer is that that this is the push, right? Is you'll notice that a lot of studies now say we're doing multi-omics, which drives me crazy. I roll my eyes because we're going to do proteomics and metabolomics, and we're going to do this, and we're going to look at the microbiome, and we're going to look at genomics, and probably all that's relevant, but who has the funding to do all of that well and organize that? And so I'm more of a reductionist, I guess, than I probably want to be, but I tend to say I got to look at what other factors. The clinical factors alone don't explain the biology that underlies this, right? We can't, when you see variability across age ranges, when you see variability in exposure causing differences in the outcome, right? Some children are exposed to higher doses and don't have hearing loss, and some are exposed to low doses and have hearing loss. Those suggest something beyond the clinical factors that we have available. And so I think about genomics as just an added element of doing that. And then to link with people like me to talk about what would be required to do that. And I think I was just talking to Jeff about this in academia. We've created scientists that we say be competitive, be collaborative, and be independent. I don't know how to do those three things together.
Yeah.
And so what we need to do is we need to figure out in a multidisciplinary way, what are the factors that we should look at? I wrote a grant some years ago with my friend Dan Bernstein at Stanford, that the NIH trashed. It was like the worst reviews I'd ever gotten on a grant ever. They hated it. And I talked about looking at extreme phenotypes. This is where I think the real value is. Extreme phenotypes are the patients that are extraordinary responders and those that seem to have toxicity in ways that just doesn't make sense, right? Like you can think about a cisplatin exposed patient who gets a fairly low dose of cisplatin and ends up with hearing loss, or the patient who dramatically responds to an antidepressant when in fact most don't have dramatic responses.
Sure.
Those are the patients I really want to study, the extreme responders, and compare them to the opposite, the patient. I want to know about the best genetic comparison would be the extreme responder to an antidepressant to an extreme non-responder. That has absolutely no effect at all. Like those are the patient's genetics that I want to understand. What makes them different, right? What makes us all different? And that's the part that is increasingly difficult. You can get overwhelmed easily because we carry, based on ancestry, we carry these traits, genetic traits differently, right? And we are still learning how to understand that. The genetic diversity of the continent of Africa is probably greater than the rest of the world combined.
Probably.
And yet we talk about, Africans. Drives me
That's
crazy.
one, yeah.
Yeah, right? Because there's been millions of more years of human evolution there. And that's why the genetic diversity is so rich and understood. And that's why we need to involve, this isn't, this is equity and diversity, right? It's about trying to understand that rainbow that exists within that continent and how that affects. And then you've got all of the people that, humans migrate. We have since humans began on the earth. And so because of that migration, we're admixed even more, right? And so there's even more diversity as time goes on that we have to explain. And so I think that for pharmacoepidemiologists, when there is something you can't explain, the question is, what else could I look at? This is fairly well-developed methods. There are other omics that you can look at, but I get a little bit nervous when people say, well, we'll just do everything. We'll collect DNA and we'll do all of the studies. Really? Where are you going to get the money for that, right? Well, and then they end up being networks that collect samples to hopefully use in the future. But then, it's like real world data. If you don't have the right outcomes, 10 years from now, you're going to be scratching your head saying they didn't collect the right data. Every time we create a case definition for an adverse event, we a priori decide what's relevant, and then we start collecting the data and we change our minds. We end up adapting and developing that further. So that's where I think this needs to go. And I'd welcome potential collaborations, even some discussions about how to work together and what problems we should tackle in genetics would really help, I think, particularly rare disease communities understand how best to treat the patients that exist and how best to manage them.
That was the most robust answer I've heard from a question. So thank you very much. That was quite elaborate. Thank you.
Too much. Thank you. Are you connecting? No.
No. Okay. I think we can do this. Or maybe not. Hang on.
Yeah, there you go.
There we go. Okay. So as I am pulling up my slides, whoops. Or maybe not. There we go. I'm going to make a quick comment. Bruce always tells me that it's so much better to study kids and children because they're cleaner populations. They haven't been exposed to a lot of things that adults are, and I deal with adults. I'm an adult oncologist. But I gotta tell you, this year when you presented this information about maybe it's the dose intensity for cisplatin, I laughed inside because in adults, unlike in pediatrics, we don't have so many different disease sites, sarcomas and so forth, all treated. In fact, you and I are on the same paper dealing with ototoxicity in head and neck cancers. And there we actually discovered exactly this, that if you give the same total dose spread over as weekly low dose cisplatin, you have a 4 times less chance of developing ototoxicity. That's a paper we're on together. So I would say in that case, the adults actually are much easier to deal with because we don't have the heterogeneity of the various treatments associated. It was just you have choice 1 or choice 2, high dose or low dose weekly in that case. So with that in mind, I'm going to talk about something different in this case. So I'm an adult medical oncologist. And unlike all the others in here, I'm extraordinarily conflicted from here. I'm going to start off with a case study because as an oncologist, this is what I do. I have a 58-year-old woman, previously healthy, is on rosuvastatin for cholesterol, diltiazem for high blood pressure, was coughing for 2 months and not responding with antibiotics, a lifetime never smoker in this case. A chest X-ray led to a CT of the chest, a PET scan, MRI of the brain, and has bilateral lung lesions and mediastinal lymph nodes that are enlarged. Received a needle biopsy and was diagnosed with adenocarcinoma, consistent with a primary lung cancer. So this is the slide that I deal with, where it's not germline pharmacogenomics that I deal with, it's somatic pharmacogenomics that I deal with. So patients that I deal with, the first thing that I deal with as I deal with lung cancer is this something that is potentially targetable because there is a genetic mutation or molecular alteration that is linked with a treatment that really works well? And by working really well, I mean things in the order of overall response rates of 80, 90%. Duration of responses with metastatic disease of 30 months, 35 months, that sort of thing, relative to standard chemotherapy, 6, 7 months of response, 25 to 30% response rate. So I deal with something in which it is very well known that if you can find a target, you should target it. And I must say that I think this is leading to drug companies making lots of money, because if you look around that pie circle, those are all some of the drugs that are available or in development. And every year there are more and more. And even if there aren't, there are all these new antibody drug conjugates, immunotherapy, cell therapy that are involved. And as an example, this right here is the weekly Canadian Agency for Drug and Therapeutics and Health. That's the agency that actually reviews for potential public funding in Canada. And if you notice, there are really two large categories. This category called non-oncologic drugs, and this other category called oncologic drugs. So they're about equal numbers if you look throughout the year of this. So cancer takes up about half of all the new drugs and drug submissions that are coming through, at least in Canada, and I'm sure in other places as well. So I'm going to talk a little bit about 4 different areas. So my focus is different in the sense that I am giving an extension and pragmatic examples of what Donna had spoken about early on. So you'll see how the theoretical that she describes comes out to play when I give some practical examples. And the first that I'll talk about in the 4 areas is kind of understanding the role of biomarkers in its adoption. And so this is also an extension of what Wei had been talking about as well. So this is the requisite slide that I won't go through that basically says that when it comes to immune checkpoint inhibitors, the main source of immunotherapy that we deal with in lung cancer as well as in many other solid tumor sites, we use markers such as anti-PD-1 and anti-PD-L1 in order to actually measure the potential for response to therapy. There are other ones as well. There's microsatellite instability or defective mismatch repair, and tumor mutational burden. And there are other ones that are on the horizon as well. So there's a whole host of these, which I won't go through. You can look at the slides. They've been presented for you to download as well. The bottom line is none of these markers are particularly that sensitive or specific in comparison to what we get when we have an EGFR classical mutation, and the response rates that we get are 90%, or an ALK translocation, for instance, in which it's close to 100% response rate. So the bottom line is these are okay markers, but they're not perfect by any means compared to what I'm afraid to say, we've been spoiled in many of our cases. But there are some issues, some pragmatic issues that need to come out. And one of them has to do with measurement of outcomes. Because there's a new series of outcomes that are associated with these immune checkpoint inhibitors. They only occur in maybe 5 or 10% of the population, but they can screw up your analyses unless you think about them or take them into account, especially when it comes to real-world data. One of them is hyperprogression and the other is pseudoprogression. So hyperprogression is what happens when you actually have a tumor that's growing at this rate. And the moment you start the immunotherapy, it just sets off a firestorm and it starts, the steep slope of growth suddenly becomes like this. It's awfully scary to see it. I saw it in a patient a week ago. The patient died yesterday. This is how rapid we are talking about. And you know, by the time they showed up in the emergency room, they had a pericardial effusion that was massive. They tried to do a pericardial stent in, and the next thing you know, that they couldn't get through because the tumor was so thick. It was literally growing day by day in here. And so there are different definitions of hyper-progressive disease. And I don't want you to memorize or learn this because it'll change every 6 months. The bottom line is you need to take these things into account, which means you can't do or design studies without talking with clinicians. That's my point in the middle of all of this, because these things are changing forever. This is another one. This is pseudo-progression. It appears that the disease is getting worse after you start the immune checkpoint inhibitor. And this is not specific to immunotherapy, but it's most commonly seen with immunotherapy. It looks like it's getting worse, but the patient feels fine. And you decide to continue treating them. And 7 months later, a year later, the tumor is actually shrinking. So what happens is that there's an inflammatory response that occurs with the immunotherapy. After all, the immunotherapy kind of drives up your immune system. What I describe to patients is it revs up your immune system to attack the cancer. So is it any surprise in some cases you get lots and lots of white cells, lots of inflammatory response, and on a CT scan, they look identical to disease progression. So these are things that you have to kind of keep in mind, which also leads to this aspect of continuation after progression or treatment after progression. We see that with targeted therapy, we see that with immune checkpoint inhibitor. And you can start thinking in your mind how difficult this might be to collect data in the real world setting when these things are not necessarily expressed in the text in a synoptic fashion, and sometimes not even expressed fully in the notes, right? So those are things to take into consideration. So one of the things you can do is to plot the growth of these tumors. And then you might see in some cases it grows and then it shrinks. That's pseudo progression. You see it sound like this and then starts to go crazy in terms of growth. That might be hyperprogression, for instance. So spider plots are particularly useful for tracking this. Great for clinical trials, really difficult to do in the real world setting. Okay, I'll take questions at the end. So there have been tools that are being developed. Our center is developing them. There are companies out there that we're collaborating with. So I know there are tons of places that are trying to either look at the natural language processing of the reported notes, or actually going back to the original imaging and trying to determine what actually happens with these tumors. Stay tuned because I think it's very, very early stage right now. And I won't go into all the challenges, but there are tons of challenges associated with this, not the least of which is the fact that the reporting is not uniform. And you know, what happens if you're segmenting a particular lesion and there are 10 lesions, right? And they're all doing different things. So those are all issues that we're contending with right now. I'm going to give you another pragmatic example of things that you need to be careful of. For instance, when we're dealing with identifying a target, how do we identify it? Sometimes we would use immunohistochemistry, right? So ALK and ROS1 are IHC based. But then for ROS1 in particular, it's not specific enough. So you have to confirm with fluorescent in situ hybridization of fish. So you can do that. Or many of us are now switching over to next generation sequencing. So you would think in the future, this might not be an issue, but many of us are still living in an era where we're looking at data in the last 5 or 6 years, where the transition hasn't occurred yet. What you need to realize is that not all NGS or next generation sequencing is the same. you could actually run an entire NGS panel and miss the one gene that you're interested in. All right, because each platform is different. So you have to collect information about the platform and the version of that platform. So in other words, Oncomine Comprehensive Assay 3.0 is very different than Oncomine Comprehensive Assay 2.0, for instance, as one example. And the other thing is that if you're looking at the molecular alteration, if you're dealing with fusions or translocations, they are best captured using RNA rather than DNA. Whereas point mutations, for instance, and deletions and insertions are easily caught using DNA. So sometimes you might not realize, and you order a test. And for instance, Oncomine Comprehensive Assay I use as an example, but there are Foundation Medicine, a whole bunch of other ones as well. So when you're looking at the tissue, they run a DNA and an RNA panel, and if you're not careful, if you're not collecting information, sometimes about 5% of the time, the RNA panel will fail more often than the DNA panel. And you don't realize that you're missing the fusions when you're collecting that information. So you need to realize that testing strategies differ from place to place and definitely globally as well. So this is another pragmatic issue you need to think about. You need to talk to the locations in which you're actually doing the studies. And if you're doing kind of a multi-institutional or a multi-country study, you'll have to look to see how that information is captured. We're going to move on to comparison of standard of care treatment arms in the real world setting versus control arms of RCTs. So something like an external control database, for instance, but one designed to compare with a phase 3 trial, that the control arm of a phase 3 trial, for instance. And I'm going to give the example of ELK, because if you have a driver, you should basically try to target it, right? In this sense. Okay, here is another pragmatic example. This is in the year 2020, so not that long ago. If you do a retrospective control study, this could easily be something that you would have captured this year. So at that time, we had a report of a patient that clearly at the bottom says there are no biomarker or genomic findings for this sample. And yet there's a RET rearrangement. And up on top, right, you see variants of unknown significance. It was stuck in the, we don't know what this means category. 9 months later, so in the meantime, right, like RET rearrangements, if they are considered functional, there are drugs available. One of the drugs is selpercatinib. Right, there are other drugs as well, but that was the one that we had access to at that time, right? And in the meantime, Lilly had announced that they had, a drug, for instance, that had a 68% objective response rate to this. So we went ahead and put this patient on the drug, even though officially the report was, we don't know what this means. All right, and if you were to collect the data retrospectively, you would see that it wouldn't be in the functional significance category. About a year later, there was an update of that report. They sent a new report to say, oh, wait a second. We've now discovered that those 2 breakpoints and that variant of unknown significance is actually significant. So, and not always do we get reports like this. We just happened to look at it, and I found it completely by accident, because for some reason I was showing a resident, that we didn't have. We went ahead, we did this. She's doing marvelously, and then I opened up, and then it said, oh, wait a second, we've changed our mind. This is now a significant fusion mutation associated with this, right? So can you imagine how difficult it is to capture this from in electronic health record, for instance? So those are things that you need to keep in mind, the fact that over time, things can change. So I'm going to quickly talk about and give you an example. And this is, I'm going to use Flora. So if you're comparing with a phase 3 trial, for instance, you basically want to mimic, you want your real world data to mimic the eligibility criteria and the outcomes of the phase 3 trial, for instance. So that means collecting key inclusion criteria, right? So you want to know the age of the patient, you want to know the performance status. Not that easy, I can tell you, to collect outside of a trial. Most oncologists don't bother saying that a patient is ECOG performance status 1 or 2. That's a measurement of how functional they are. Where the idea is that 0 is that they're completely normal, and 4 is they're completely bedridden, and 1, 2, 3 are somewhere in the middle. All right, but that's a requirement for a trial like this. In the real world, difficult to get. We need to know and confirm the mutation, which is exon 19 deletion L858R. That's the third blue arrow in there that we can get from the reports. They have to have no prior systemic therapy or that we can collect. They need to have stable brain metastasis, and they will be allowed, that we can collect. But already you can see performance status becomes a bit of an issue for us in this particular setting. And then we have to collect some standard variables, right? We need to have the staging data, the metastatic profile, prior treatments, in some cases, viral status, for instance. That's probably one that probably isn't that important. But again, if you're trying to mimic what was done in the trial, you've got to see what you can collect, and you collect as much as you can. And the performance, as I said, was going to be difficult. And then the molecular and biomarker tests, right? So staging is actually more complicated than we realized, because in the middle of that study, for instance, the staging systems change from the 7th edition to the 8th edition. So exactly which definition do we use in that particular setting? So that's something that one has to figure out. And we use pragmatic choices, right? Sometimes we use the existing, however it was staged, because that's real world data. And sometimes we have no choice, but then to try to see if we can correct as much as we can to the latest staging system, or the one in which the trial was performed in. And then we have to collect all the outcomes data. So these are all the other blue arrows. So progression-free survival was the outcome for this study. Objective response rate, and as mentioned by Donna, really difficult to get in real life, in the real world. Duration of response, I'll explain why that's also difficult. Disease control rate, difficult. Depth of response, oh, don't even go there. Overall survival, easy. That's live or dead. Thank goodness for something, right? Patient-reported outcomes, probably not available, right? And safety, which I won't go into because Bruce talked a lot about toxicity already. But again, difficult to collect unless it is so meaningful that it causes a dose interruption, a dose reduction, or permanent discontinuation, right? Those are kind of the standard ones that we would talk about in a clinical trial in this case. So then we start looking to see, do we have standardized elements that we can use in the real world to mimic what goes on in trials? And the two large groups, there's OMOP, the Observational Medical Outcomes Partnership Common Data Model, but that's not specific to cancer. A number of cancer associations, like the American Society of Clinical Oncology, the European Society of Medical Oncology, and other organizations have rather preferred to use the Minimum Common Oncology Data Elements, or MCODE, in this case. These are all the various, different types of data elements you can collect with OMOP, which is the generic one. And these are the ones for MCODE, right? Lots of things to collect, wonderful. But if you look to see in terms of how much detail they give for the outcomes, that's it. And that's because outcomes are really heterogeneous. And you have to define them according to what your needs are in this particular case. So as an example, progression-free survival, we're going to have to use a pragmatic definition for this. So if a clinician says, oh, the patient has progressed, we got to go with it. No one's going to go back and remeasure all of this using RECIST criteria, for instance. Then in some case, but there's an assumption, right? Progression-free survival assumes that if you died without having the disease progressed, that is an outcome. And so that's particularly useful for advanced disease, where the more likely scenario is that when people die, they probably died of their disease, even if you haven't captured the fact that the disease has gotten worse. Time to regression is kind of the opposite, where you censor it. So in other words, you assume that if someone died without an event, that they didn't die of their disease. So that's a little bit more useful with early stage disease, for instance. So those are things that you kind of have to think about when you're deciding what to use. And this is RECIST criteria. This slide, so I deal with RECIST criteria practically every week because I'm the one who's actually making the assessments for patients on trials. that I run at my center. And I can tell you that I have to look up things probably once a month anyway. I've been doing this for 16 years. So in other words, if it's that difficult for me, imagine how difficult it is for even a machine learning program or something to actually deal with. So we don't use RECIST criteria. We use the closest thing. So we put an RW in small letters before all of these outcomes. RW, progression-free survival. RW, duration of response, right? Because that's what we're using. So fundamentally not possible to really do accurately. We have to use some other things as well. So what are some of the other variables that you use? So time to first subsequent therapy or time to treatment discontinuation are perhaps reasonable surrogates in some circumstances, but not all. All right, to actually use in place of progression-free survival, for instance, in different settings. Not exactly the same definition, but at least you're able to define it very clearly up front. So that would be, I would imagine, a discussion with Dr. Rivera and the FDA in terms of what would be reasonable for RWD data to collect. So, and finally, overall survival, I said, that's the easy one. So, yeah, it's really hard to get it wrong. I guess it's possible to get it wrong, right? For people to have died and not died, but it's probably the most accurate real world data element for outcomes for now. I'm going to talk about comparison of standard care treatment arms with single phase 2. This is exactly what Donna was talking about when she was talking about the external control database in this setting. So the example I'm going to give is with entrectinib and ROS1. So ROS1 rearrangements occur in less than 1% of all lung cancers. They say 1%, but we've never been able to demonstrate that 1%. It's always less than 1%. So it's pretty rare in this case. And so the entrectinib data all came about not from randomized controlled studies, but from single arm studies. So maybe in the US you can get away with this, but in Canada, we have to determine what the financial benefit is, right? So we have to do an HEOR, a health economics outcomes study in order to determine this. So when you don't have a control arm, you have to kind of quote unquote find one kind of along the way. So in places like Canada, we cannot approve any of these drugs without some of these analyses being performed anyway. So in this case, a group used the Flatiron, which I think all of you are aware of, and looked at their structured data and then tried to deal with the unstructured data and process it in one form or another. So what they did was the actual entrectinib data was crunched together from 3 entrectinib single arm trials. In order to get a decent number of patients, and then compared with a crizotinib arm that was the control arm for Flatiron data in this case. So in order to do this, you can imagine they needed to balance the baseline characteristics, so they used propensity score based balancing. They looked at weighted time to treatment discontinuation and overall survival data. Like I said, overall survival was pretty safe. They decided to use a surrogate for progression-free survival, which I think was reasonable in this particular case. And they found a difference. And in fact, this was data that had been submitted to Health Canada and helped to actually lead to the approval across Canada, except one province, for entrectinib. That one province hasn't approved any drugs in years. So in this case, they were also looking at potentially duration of response, but again, difficult to obtain from real world data. So that's why they they didn't really discuss this to a great extent in this study. They talked about duration of treatment, which is a much easier outcome to collect than duration of response, because response implies that you're tracking until it grows to a certain size, usually measured through RECIST criteria. The complication is that there's treatment beyond progression. So many of these targeted agents, and even I said some of the IO therapies, there's treatment beyond progression. So when you're talking about the duration of treatment, sometimes the patients progress, but you just kept them on. Pragmatically, it probably doesn't matter, but if you're comparing it to a trial data, it does matter, because the trial would clearly have time to progression or progression-free survival, right? So it's a slightly different definition. One of the things that we deal with in this case is use other types of graphics, like swimmer's plots. So we can identify, for instance, and many of these are rare tumor subtypes, for instance. So the swimmer's plots won't be, 1,000 patients, that sort of thing. Where you can actually see at what point they failed or progress, as far as we can tell, the RW PFS, and then the post-progression treatment, and collect data on both, for instance. So those are things that you ought to be thinking about as you're designing some of these data if you're using it for this purpose. All right. And so this way we can collect information such as treatment beyond progression and still be able to actually visualize it to some extent. All right. Waterfall plots are useful to describe the depth of response to therapy. We are seeing a lot of these for targeted therapy, for instance. But again, this implies that you can measure how much the tumor has shrunk or how much the tumor has actually grown. And these are really difficult to do without completely going back to the original data. And we're trying to do this with NLP, and we're trying to do this by actually looking at the original imaging. But you can see, right, how difficult this is, because for large scale studies like Flatiron, you never, that isn't, the NLP maybe, the natural language processing maybe for the reports, but definitely no one's going to go back to the original scans to actually look at this. Okay. So the last section points I want to talk about has to do with understanding the selection of different treatments based on patient characteristics. And that can be quite useful as well with real-world data. So as an example, for the ALK rearranged lung cancer, how do we know that this is a very different population? Well, the risk factors are different. They occur more often in never smokers, although it hasn't been really published to a great extent. In places like Toronto, for instance, where we have very ethnically diverse groups, we see this much more often in non-Caucasians than in Caucasians. So we think there probably is a predisposition. The median age is younger by 10 to 15 years. It's a single driver oncogene as opposed to most lung cancers where the idea is you have multiple DNA damage hits that lead to the cancer itself. The resistance mechanisms are totally different. They're usually in ALK related to one of two things, either another mutation that occurs in the gene that now makes the current drug useless because it can't bind or can't fit because the configuration of the molecule has changed, or bypass track. So in other words, instead of going down the ALK pathway, it bypasses it and goes down the NRAS pathway, for instance. And the prognosis is different because unlike a median overall survival of about 14 to 16 months, a median overall survival from the date of metastatic diagnosis in ALK is 6+ years. So you can see very, very different in its nature. So you can't compare apples and oranges in this case. So where do you look for some of this data? You could look at very large cancer registries and administrative databases, such as the French has a national database that's been very useful. Flatiron was one that I mentioned before. They're great resources, but most jurisdictions won't have them. There's a lot of infrastructure building in order to get there. And there's still, despite the fact that supposedly Flatiron data just magically comes out, there's an intense amount of manual work that's done in order to actually get it to a point where it's interpretable. And or you can do multi-institutional single marker-based studies, right? But they're costly, and you're doing it like a one-off study in this case. So doable, but quite expensive. So we were thinking over the last number of years of taking or stealing from the clinical trials world, where they have these things called basket and umbrella trials. So an umbrella clinical trial is one in which you have an umbrella, let's say, of lung cancer, and then you have different subgroups in between, and you run one study, but each of the subgroups is actually treated as its own sub-study. So you might randomize, if you have an EGR mutation, you randomize to treatment A or B. If it's ALK, you randomize between treatment C or D, etc. Or a basket trial in which you have different diseases with the same molecular alteration. We do have some molecular alterations that cuts across disease sites. So NTRK would be an example of that as well, where we see across about 20 to 25 different disease sites. So what we did was we said, well, if they're able to do these types of clinical trials, then they do them because they're efficient. They're efficient because they have a common infrastructure. There's less infrastructure. There's more clinician buy-in because they open one study instead of like 8 studies, right? And then when one arm closes and another one opens, it's an amendment, which is a lot easier and a lot cheaper than going through the whole process of a new study. The coordinators like it because they're collecting common bits of information where they can. In some cases, they're very specific to the arm, and that's different. But a lot of things are common, so they get used to it as well. The arms are expandable, and different arms can be funded by different pharma companies or peer review grants or something else. So we thought, why can't we do the same thing but just not have the randomization? We just observe. So this is an observational study. So we started calling it a bureau, so a basket umbrella real-world observational study in this case. And so that came about for us as a Canadian Rare Molecular Alteration basket umbrella real-world observational study. Shorthand form is CARMA-BROS, much easier to say, and it is actually registered as a clinical trial with an NCT number, for instance. So and so it currently is supported by 5 different pharma companies, and with 8 different open arms and more to come in this case. And I'll walk you through a few things about it. So we'll try and describe what is a bureau study, and we realize it really is an adaptive study and a universal platform for which we can build these cohorts for real-world observational study. We designed it initially to capture lung cancer, but we're actually capturing other cancers as well now, because the L for lung cancer does not appear anywhere in the abbreviation for CARMA-BROS. So it can be expanded to cover a whole bunch of other things. And on the slide, right, we know that retrospective chart reviews, multi-site single marker registries, single site registries on the bottom left, they're less universal and less adaptive. It's a one-off, right? And the same with kind of cancer-specific registries in that case. Then there are some that are more adaptive, you know, adaptive clinical trials and so forth, and some that are more universal. So our ALPRO study, our adaptive universal platform, now also is used in some cases to collect tissue samples, to store blood samples, for instance, right? So the universal nature of collecting a whole bunch of different data and data elements, I think makes it useful in this setting. So right now, on the top, you have the list of open arms that we have right now. And then we're just slowly moving to GI cancers now. We're going a little bit steady by steady right now. Our data has actually been accepted by the FDA, EMA through UK, and EMA through France, Bulgaria, and China, of all places, and Health Canada as well. So the data elements are reasonable, and this seems to be a reasonable design. We often will do ambispective studies because we don't want to miss have the issue of missing dead patients, right? Along the way. So we'll include retrospective, ambispective or prevalent cases, and prospective incident cases as well. And whenever we could, we sometimes can collect patient-reported outcomes as well. So it took us 5 years to develop this. It's open at 16 sites across Canada, which is not bad because there are under 20 cities that are considered fairly large in Canada. So we've got 16 of them, and 7 sites are waiting to open right now. And this is just a little bit of data that was just published last year from the work that we're doing. So we looked at the Canadian ALK experience, for instance, and we showed that to get people to live up to 10, 11, 12 years, you need to have access to multiple drugs. All those pastel colors, each one of them on the swimmer spot. A swimmer spot is when each row is a different patient, right? So different pastel colors means the time in which they're receiving different drugs. And this is important because right now in Canada, we only have approval for one drug. You can choose which one, but you only have access to one. And we're going to use this data to try to argue that they need to allow subsequent treatments and subsequent tyrosine kinase inhibitors to be used in this setting. Otherwise, you can't get them to live 8, 9, 10 years. I don't know if you can see at the bottom, but the scale goes up to 12 years there. So what we argue is choices need to be available. Often patients are on the same drug for years, so toxicity becomes an issue, right? And many patients can live a long time, but only if they get that tapestry of different drugs kind of along the way. And we also can see there's a subgroup of patients that all die in the first 2 years. So even though we have a median overall survival of 6 years, there's still a subgroup that there's an unmet need for. So all of this we're able to actually determine, and this is what we're going through. We're able to determine where these cancers spread. So these are the cumulative rates of metastatic sites like bone and brain and liver and lung and so forth. We're able to demonstrate that there's prognostic value in the presence of different metastases at the time of diagnosis, for instance, like liver and bone mets argue for worse prognosis. And we've also collected quality of life health utility data in this case, demonstrating that as long as they are stable clinically in the real world setting on these drugs, their health utility scores are between 0.8 and 0.9. My health utility score is somewhere between 0.8 and 0.9. So I think that's quite decent in this particular setting. And then we're able to actually, unlike the trials, you can compare only one drug with another. We're able to actually do this across all of these drugs. So we can then say, you know, green means lower toxicities, and red means greater toxicities, with a list of toxicities in the rows and the list of different drugs in columns, for instance. So we're able to also talk about treatment modifications in here, whether there are treatment delays, dose reductions, or permanent discontinuations. So my take-home message with all of this is simple, that oncology data elements include specialized variables that you really need to think about when you're designing these studies. And they're different from disease site to disease site, from type of drug to different type of drug as well. Some variables are not routinely captured in real-world data, so you need to think about alternative outcomes and the pros and cons of selecting each one. You may need to consider either OMOP compliant or MCO compliant databases if you want to then think about submission into a regulatory agency, for instance, and also think about some of the novel types of graphical representations, such as the spider plot, the waterfall plots, and the swimmers plots, where you can determine with exactly what Donna said, multiple epidemiologists, statisticians, clinicians, right? Which variables are critical versus preferred versus not, especially when it comes to confounders, the eligibility criteria, and sensitivity variables that you may want to use in your sensitivity analysis before you start your RWD analysis. And I'm getting a nod from Donna, so I know I'm saying something right. And finally, consider the jurisdictional registries, these all-pro study designs, these hybrid new types of designs that might be quite useful in the real-world setting. And hopefully in the future, we may be able to validate use of natural language processing or other tools right now, which I'm pretty certain would not be considered acceptable or standard at the moment. All right, and finally, the oncology observational studies often involve smaller sample sizes. Like once you cut, lung cancer is the most common cause of death, and it is obviously, if not the number one, often the number one or number two incident case. But once you divide them up into little categories, they suddenly become rare cancers, for instance, and that's becoming more and more the same with breast and renal cell and prostate. They all have molecularly or biomarker defined subgroups. So with these smaller sample sizes, due to these biomarker driven therapies, they raise issues about the lack of standard phase 3 trials in some settings and the need for this type of data, if it's done in a quality fashion, could be very useful to document effectiveness in the real world. And I'll stop right there. Thank you. Are there any questions? Okay, thank you.
So thanks, Jeff and Bruce. Those were great presentations. So at this point, I want to invite all the speakers up to the panel, and we're going to have an open question and answer period. And no question is off limits. So if Jeff and Wei and Donna, Bruce, and I've got some questions if other people don't have questions. So why don't you go ahead as Jeff is getting some water. After speaking for 30 minutes straight. So go ahead.
I'll wait. My question is for Jeff, so I'll wait for him to say.
Oh, yeah, no, go ahead.
All right. So first of all, thanks everyone for this great talk and bringing everything together. It was very enlightening for me. Pseudo progression. So I have two questions. Pseudo progression, can that be in the other direction? Meaning, do you see patients who initially seem to be improving, and then after some time everything goes downhill? And is that
wait.
pseudo progression?
It's already on.
I don't have to push it.
Yeah,
Okay,
yeah.
all right. The bottom line is when that happens, this is what we expect of most of our stage 4 patients, because at some point they're going to fail. In this particular setting, right? Where the problem occurs is you don't want to stop a drug that could still be effective, right? Because you think things are getting worse. In the other case scenario, it generally is not so much of an issue because, you know, if it shrinks, you know, do I care if it's real or not? No, it's not growing, right? So for many of us, stability or shrinkage is what is clinically relevant for us, right? For the trialists, shrinkage is really important because that's where you get your response. But disease control rates are, I think, which is a combination of stable disease with responses, are I think very important for patients, for many patients in that case.
And then this question, thanks. This question is for the whole group, but in terms of thinking of time to, I think you call it time to subsequent therapy or time to, how do we standardize that in claims or EMR data? So if I take, say, a common claims database in the US, for example, there might be different regimens or approaches from center to center. And so the factors influencing initiating a new line of therapy, you know, may not always be related to progression. So any ideas or thoughts about using time to new line of treatment as an outcome in real world, and how to standardize that in view of center to center differences in treatment regimens?
I can maybe start that, and maybe my colleagues can comment further. This is a big issue for a couple of reasons, and you've hit the nail right on the head, which is there are many reasons why someone either starts or doesn't start the next line of therapy. When you're looking at that, you also have the problem, the fact that some people never start another line of therapy, and you don't know why. Is it because they're getting worse, or they're traveling the world, or they've left the system, right? And so they move to another HMO, for instance, right? And so we run into those issues all the time. Then there's the other aspect, which is time to discontinuation. So there is the discontinuation of one drug, and then kind of waiting until the initiation of the next one. So sometimes we have to actually report both, because they're for us kind of the two extremes. In this particular case, and then you still have to define what happens if you think they're lost to the system, right? And you obviously have to define death as an event, presumably because they can't continue to take drug if they're dead.
I might add, oh, do you want to?
Sure,
Go ahead,
Oh,
yeah.
Yeah. So I agree that this is a significant methodological question in the context of real world data. And I think that, you know, you have TTD, time to treatment discontinuation, TTNT, time to next treatment. And those are actually outcomes that in some ways, I think people think are potential proxy to PFS, or, you know, types of kind of understanding of progression. And there's actually been some work done on that there are some correlations. I think the concern is exactly as you are suggesting, which is what do you deal with when you don't actually completely understand why there was, you know, this TTNT, and to your point, one of the questions around this is actually something that the Friends of Cancer Research is addressing in their current pilot. So the Friends of Cancer Research has a pilot with 7 data groups that's working on addressing and understanding real world response. So thinking through how to elucidate real world response, and their previous pilot actually looked at some of these other types of outcomes to understand. And what they did is they created a standard set of rules that each data set would apply to these specific, looking at say 180 days and then trying to figure out if we could apply a standard rule set to look at time to next treatment. And if there's not a treatment in that window, then you count it as, not part of that outcome. But I think that being said, I think it's still, even if you apply a standard set of rules, it's still challenging for exactly the reasons that Jeff mentioned.
I agree everything they said. Probably just add several perspectives because we are currently actually doing some claim analysis. Two data set, one is Optum another one is marketing scan. You are exactly right. The claim data set is more challenging than EMR. Part of the reason is also the coding may not be validated or developed. Sometimes you have to develop the coding. It's very important. Secondly, that's really to previous kind of emphasized by Jeff. You have to work with a clinician very closely. How do you define a regimen? Because they may starting with one drug, add another one in the next couple months, whatever. So how do you, is starting a new line of therapy with still the same line? So that's where some of the clinical judgment is very important. So work with clinicians is extremely important. I just want to add those two perspectives.
One last thing is the fact that if you would try to use time to treatment discontinuation, that doesn't work if you have like a finite set of cycles of treatment, for instance. So in some cases, for some diseases, they give 6 treatments and that's it. 6 cycles, 4 months of treatment, 6 months of treatment, and that's it. So that becomes completely useless in that particular setting for that particular question. And so that's the extension of why you need to talk to KOLs, right? To folks who know the actual patients and the treatments and so forth when you're deciding what to use, what surrogates to use.
Yeah, I completely agree. Right, it's clinical setting dependent. I think you'll hear that a lot from FDA, from others, that the clinical context for what you're doing really matters. Like, for example, lung cancer, very different than breast cancer, and even in the way you think about how the patients are being treated, the modalities of treatment that are available, all of these different things. So yeah, I think it's so dependent on the clinical context.
Thank you very much for that. If we're in one healthcare system, if there was a database that was just provided by one healthcare system, do you find that there are still differences from center to center, even though it's one health system, or like the health system decides this is how we're going to manage breast cancer, everybody must do it?
I mean, does every doctor come from the same university? Graduate from the same university, have the same experiences, never move, never immigrate, never go to another state. So that's the problem, right? Is that no matter how many rules that you restrict physicians, as a physician, I can tell you, we find lots of ways to bypass rules.
What about, though, I would be interested for both of you in Canada, do you find, because I would say in Kaiser, for example, I think it's a little bit, I mean, there is still variation. The clinical practice is clinical practice, and people practice to the way they do. So, but do you find it? How is it in a country that has nationalized healthcare?
Well, it's not national; it's provincial, but
right,
because we don't have socialized medicine in Canada, we're like every province is its own HMO, but it's still quite variable. And I think going down the level of prescriber, I mean, the issue always is. Depends on the family too, in the case of pediatric cancer. So, we had a family some years ago now that were orchestral musicians. So hearing about cisplatin and it causing hearing loss and losing high frequencies, they were dead set against it. We brought in an ethicist. There was these big discussions about either child was going to die if they didn't get cisplatin. They refused treatment, even with the ethics people involved and so forth. That child, by the way, alive and well and cured without cisplatin and hearing. Wow. So, so I think for clinicians, if they're listening, they're also relying on what the family experience is. And none of us thought that was a good idea, and wouldn't want to repeat it again. But that's the variability. That's another factor in variability. It's not just prescriber, it's also the patient you're treating in front of you and how the families.
So for instance, if I have another available line of therapy this year that I didn't have last year, I might be more likely to switch the patient as soon as they progress. Whereas last year, I'll just keep them on because I've got nothing else. Can you see what I mean? Right? And all my notes would say, yeah, kind of stable.
But
Right.
that's what you can't measure in real world data, right? So I
That's
think, yeah,
There's no hope.
there's always hope.
You know what? At the end of the day, right? If you think of it from a pragmatic perspective, it may matter a little less in that case, right? Because that's how we're actually treating in the real world. And for some of us, treating in the real world is more representative of what we want rather than what's in a very pure clinical trial. So it may not be the worst thing in the world. It's terrible if you're an epidemiologist. So every day I put on two hats and I fight with myself.
Well, that's where the fit for purpose, right? I mean, it's like you don't have hope. I mean, it's each of the data set or study can answer certain questions. So it's important for you to be aware which research question you can address from this particular data set.
Yeah, and like on our data, from the pediatric data that we've collected over 20 years, when we first started the data collection, I wanted lots of free text area, and the non-clinicians it was driving them crazy. And yet I can tell these stories years later because we've captured that in the free text environment, right? The richness of medical records, not always rich, but often it can tell you those stories, and that's the importance of having that story, because it does tell the differences between patients in terms of what their attitudes are, what their family needs are, and how we've dealt with interesting challenges that just don't seem to be the same as others.
So while we're waiting for more questions, let me throw a related question. So, we've been working in real world data, real world evidence for quite a while. I forget when the terms started, just replacing other terms, but the new thing that the regulatory agencies are going to consider that, and Donna put up a slide of 10 plus guidances. And so my question to those in industry and academia, and then lastly with Donna in regulatory, is to what extent maybe in the last 5 years that you've seen real world data, real world evidence actually being incorporated? I mean, Jeff showed some nice examples, but for efficacy or effectiveness on the regulatory perspective. And will this really get incorporated into our regulatory framework to some extent, maybe in natural history studies or in some ways? Or is all this real world data evidence or most of it going to apply, as Jeff said, to the clinician and the patient, where they're going to be using things off label and so forth? And it's really going to inform clinical practice and not necessarily get approval into regulatory. And so maybe I'll start with Wei, and we'll go just down there.
I can provide from industry perspective. I think you are right. I think the buzzword RWE starting a couple years ago, probably replacing the big data, whatever. Particularly in the beginning, I think people were excited. Kind of seems like FDA is encouraging to do it. But then I think cool down a little bit, people realize again what the reality is. Because FDA also made it very, very kind of specific. Again, the standard has not changed, right? No matter they're dealing with trial data or real world data, it's the same standard. And Donna's slide also shows RWE can apply in a certain kind of areas, pediatric, high unmet medical need, whatever. So I would think industry is still trying to figure out what's the best use case will be for regulatory. I think regulatory is still relatively very high bar. But I would also think using real world evidence from industry perspective should not limit it to just regulatory, because how you use this data supporting clinical development, supporting biomarker development, and the last example given by Jeff to supporting the kind of marketing access, HTA kind of assessment. Those are also kind of very important. So I think that use RWE for label expansion, particularly for regulatory purposes, is a high bar, but the next tier will be as a supportive evidence for regulatory. And another part, there are lots of other use cases, again, helps development, help the trial design, and also help the kind of reimbursement HTA submissions, or even help the clinician and the patient understand the value. I think those are also very important aspects.
Yeah, I would agree with everything that Wei has said. Um, I don't live in the US environment. So outside of the US, where the moment a drug gets FDA approval, everybody starts using it and it has to be funded or else it gets sued. Doesn't happen in Canada and doesn't necessarily happen in Europe either. So I think a lot of these studies that we've been doing have been useful and will continue to be useful in order to try to get funding to occur when funding and approval, regulatory approval might be two separate entities, or when funding requires some sort of economic analysis in this case, especially since we have more and more of these single arm phase 2 trials, rare subtypes. You have to compare with something that's a current standard. So I think the real world evidence world will still be around for a long time. The question is how much will it be accepted by the FDA? And that will be coming in 2 people from now. Comments from the stone 2 people.
Well, why don't you comment and then well, no, I mean, I'm with Jeff. I think that that where we've in pediatric oncology, where we've spent a lot of time is looking at issues of how the drugs are provided. So the cisplatin dose intensity work was really an artifact of work that we weren't doing in dose intensity. We were just trying to figure out how to include time elements. Everybody was using cumulative exposure. In the case of avascular necrosis from steroids, there's another problem, which is not everyone, but I can quote at least 3 studies published in major journals like Blood that have looked at avascular necrosis in children with cancer are using protocol dosing. They're not actually looking at the real dosing that was provided. So because we're active surveillance network, we collect the exact dosing patients are getting, and guess what? Some patients are getting 50% of the protocol dosing because they get avascular necrosis. So we stop using the drug. They're still alive. They still go into remission. So again, I think that there's what I begin wondering is, are we over-treating cancer? Now, on the negative side of that, we've got wonderful survival data to support the fact that over the last 50 years, we've gone from a 25% survival rate in the 1960s to, over 80%. British Columbia is 83.7% overall today. So we've got great survival data based on that dosing, but it's not the actual dosing. The other thing that's interesting is, and I was just at a meeting with Donna several months ago in Washington at the Childhood Cancer Data Initiative. They don't collect any information about concomitantly used drugs like SSRIs and other drugs which are known interferers, inducers, and inhibitors of metabolism of other drugs. They don't collect any of that. It's just the cancer data. So, I think this is really concerning to me that we're looking at the drug dosing that they are supposed to get, not the dosing they really get. We already have a problem with adherence. Even in pediatric cancer, we have problems with adherence and children lying about the use of their medication and adherence to it. But we're even so I think that's a real issue, is that we need to be focusing on the actual dosing that the people get, and that's and in fact, oncologists, when I bring this up, are shocked. They didn't get the same doses. No, they actually didn't. And because these are serious adverse effects, they're not just, nausea or vomiting or things that can be managed with other drugs. They actually cause permanent disability. And so we hold doses and we stop therapy early. Even cisplatin gets stopped when we get significant hearing loss, or we decide not to give the last cycle. So it's really important to look at that. And I think that's going to be necessary before I want regulators to start acting on the data, that we actually have the data to support that. And what are they interacting on? The first data that was published on tamoxifen and outcomes and whether, CYP2D6 genotyping was published included no information on concomitantly administered drugs. I mean, oh my God, that's embarrassing, actually. And so I think this is a really important area. And what is often reported is concomitant drugs as a dichotomous outcome. So if you're looking at renal function, for example, they'll say they were on other ototoxins like furosemide. Yes or no? Well, how much furosemide? 1 dose? 2 doses, 3 weeks, all of that's really important. That's not analyzed. So we have a ways to go, I think.
Great. Well, so it's always exciting to be the regulatory representative on the panel. But I'm also, I know I have some regulatory colleagues in the audience, so that's nice too. And I actually really appreciate being able to hear the perspectives of others outside of regulatory as well. So I want to say a couple things. I think first, I'm cautiously optimistic, I think in general about the use of real world data. We actually have a few examples in oncology specifically where real world data has been the basis of the new drug approval. So 2 drugs, which you'll hear more about later this week, abatacept and alpelisib, which were approved in respective ways on different types of registry data and expanded access program data. And so I do think you're starting to see the potential, but again, it's in a specific clinical context for a specific reason, that a randomized trial could not be done and that it fits the population. The data was fit for purpose. It meets all of those things I walked through earlier. Adequate and well-controlled study meets our substantial evidence bar. I think what was said earlier, the bar is high. The bar is high. And I think it should be high because, especially like I think about pediatrics, kids need medicines that are, options, but they need effective options. They need safe and effective options. So it's not just bringing drugs to market, right? It's bringing safe and effective drugs that clinicians can use and patients can use. So, I mean, and especially reflecting on just the experience in FDA oncology, we've seen an incredible increase in the amount of submissions that include the use of real world data. And so I think there's a lot of potential. I think there's also, I mean, especially speaking to external controls, some of the other methodological processes, I think there's a lot that still needs to happen. I think the underlying data is key to, I mean, the data has to be of appropriate quality, relevant, reliable, fit for purpose. There are a lot of steps. And I think that's where the pharmacoepi community can contribute. There's a lot of, I think, still methodological work to be done, but I'm hopeful that there's also not just in the context of effectiveness, but other ways that real world data can support drug development, can expand our understanding of effectiveness in different populations. There's a lot of emphasis currently on several different FDA initiatives, including diversity plans and clinical trials, and then also post-marketing evaluations. So there are a multitude of ways, I think, in which real world data can support regulatory adjacent or even directly regulatory efforts, as well as supporting clinical practice and hopefully accomplishing what everybody is really thinking is part of the promise of real world data, which is creating and advancing the use of therapies to bring, an enhanced drug development for better patient care and outcomes.
Great. Thank you very much. And I'll just add that ASCO, I think I read a paper which estimated that 60 to 70% of their guidelines are based on observational studies. So we have 4 minutes to go. 1 minute apiece. What are you most excited about in oncology? It can be in the area you're working or in another area, but what is coming down the pike? Is it machine learning? Is it AI? Is it a certain drug or a target? What are you most excited about in the next couple of years? Can we start with Jeff?
With me? Oh, wow. Okay.
I want to give away a chance.
Yeah, so I'm really excited about the use of artificial intelligence in mining our data. So I think observational research will change as we get better and better with using such data. We can just continue to validate it. So I'm excited about that, as well as the hundreds of new drugs that are coming through each year and indications.
Great. Wei?
I'm excited. Actually, again, the precision medicine or use the kind of target kind of therapies will continue to advance. Not just to help the drug development approval, but also in the reality when you get approval, how we can encourage the patient to get the testing, and therefore can guide their treatment and the next steps. Again, one of the very surprise finding actually we found is that you would assume with so many targeted therapies, a patient should almost 99%, 100% patient are going to get the genomic testing, therefore they can get the treatment, which is not. Probably Jeff, I think in your hospital, you much higher average, probably like a 50-60% of the patient, which is not just how good the drug is, it's also reality how we apply those in the real practice.
Great. Donna?
Well, at first I'm excited to be here. I think this is always one of my favorite conferences and it's really great to be able to interact with all of you. But what I'm excited for the field, I think is the expansion of the real world data paradigm. So I think I alluded to it in my presentation, but pragmatic clinical trials, I think is a really unique opportunity for epidemiology to merge with clinical and biostats and really, and even trialists, and really think about how we can pragmatically advance evidence generation and work towards building this, learning healthcare system as it has been envisioned for many years. But I think given, especially in the US, our leadership at the FDA, at NIH, that there is a unique opportunity, and also through Cancer Moonshot to maybe start catalyzing this force. And so I think I'm really excited about what pragmatic trials may mean for the future of patient-centric drug development, and also the ability to convene this in an international way. I think even especially through ISPI and other ways, there's a lot that internationally we can learn from each other. And so I think the direction in which international collaboration is going as well will hopefully build a lot of work for us, but also hopefully a lot of benefit for patients.
Great. Bruce?
Okay, so for me, it's induced pluripotent stem cells from patients where we find genetic targets of interest, and we find these proteins that are high value markers. And what we're doing now is we're drawing peripheral blood from these patients and we're growing cells that are relevant to toxicity. And I didn't get to report these data, but we did some work that's published in Cell Stem Cell and Circulation last year, where we withdrew blood, we grew cardiomyocytes, and from that we showed that we could induce that these patients with these genetic risk variants, their cell death was quicker. If they had protective variants against cardiotoxicity, their cells lived longer. That was the first thing, validated the genetic marker's value with a cell model. The next step then was to actually take that protein and develop a three-dimensional structure, and using computer-aided drug design, determine what commercially available compounds could help protect, could actually help promote that target. And so we can actually confer the protection that some people have to everyone. That's the really exciting part, and using commercially available compounds. And we just published a paper showing that dizipramine, at least in the cellular model, can actually protect the heart against anthracycline cardiotoxicity in children. We looked at rapidly dividing cells as well, and to see if that affected the viability of rapidly dividing cells, which we don't want to do because that's cancer killing. Didn't affect those. So we're at this stage now of what's the next model system that we develop before we actually try this in patients. And probably an animal model like rat or mouse will allow us to begin thinking about this. And so it brings up all sorts of really interesting questions in terms of 3D drug design, and then inducing from the cells of patients that have these genetic variants, these different cell types. It's very exciting.
Great. I want to thank all the speakers and the panelists. Terrific presentations today.